dear little champions, let's see if the live has officially started or not. Just give me a second. Yes, it's on. So, hi my dear children, my name is Jasmine Joy for those of you are asking, and ma'am, are these going to be the same MCQs from the 1 marker videos? Yeah, it will be 1 marker videos. Along with that, I have taken the model question paper videos as well. Okay, I mean model question paper questions as well, the MCQs. Let's get started my dear children. So the rule is we will go chapter wise, and I have arranged the chapter in the order of their weightage. So the first chapter, which has the highest weightage, would be Principles of Inheritance and Variation. Very big hi, Samant, Bhumi, Srinivas, all of the children, okay, Chinmay, Ashoka, all of you guys, hi Neil. So let's get started. Let's not waste any bit of your time. I know that your time is so precious. So I was thinking, if there are so many children, okay, I would go ahead, take all the chapters. If the count of children are less, okay, then we will just stop after doing most high weightage chapters. Let's see how it goes, right? Let's start off, and then only I will be able to understand the duration, but it won't be long. Okay, the first question, plants having the genotype capital A, capital B, small b will produce dash kind of gametes. First answer, how do you answer it? Write 1 and then A, B, C, D, whatever is the answer. So, the genotype is capital A, small a, capital B, small b. How do you write? Hi Riyaz. Hello, hello, hello. Hello Praju, everyone. Hi, 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 hi. Lots of people. Hello. So basically you calculate with a formula that is 2 raised to the power n. So you first try to find out the number of heterozygotes. Heterozygotes as in the one that does not look the same. So over here capital A and small a, they do not look the same. Here also capital B, small b, they also do not look the same. So n is equals to what? 2 over here. So 2 raised to the power 2 is equals to what, my dear children? It will be what? It will be 4. Got it? So your answer becomes 4. Now if it was like this, then we do not have any heterozygotes. So 2 raised to the power n will be just 2, nothing else. I hope you got the answer. So everyone says it's 4, 4 and B, 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 B. Great my dear children. Sparkle, Srinivas, Praju, Sparsha. Okay, great, 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 great going. My dear children, if you can, please write together so it will be easy for me to understand. Going ahead with the next question that we have, in a monohybrid cross between 2 heterozygous individuals, again the gametes are not same, okay, percentage of pure homozygous individual obtained in F1 generation will be. So the question says monohybrid cross, that means you're talking about just one character. Let's take it as height. Let's take it as height. Now, basically you have 2 heterozygous individuals. That means the gametes are not same. Capital T and small t and capital T, small t, these are heterozygotes. Now, what is the percentage of pure homozygous individual? That is a question, right? So, what do we have? How do we calculate that? First, we have to put the gametes. So, gametes are capital T, small t. Here also capital T, small t. If you fuse them, you get capital T, fuse them, capital T, small t. This capital T, small t, and then you have small t, small t. You are asked pure homozygous. Pure homozygous means this and this. Now, if you want to calculate the percentage, Baba, how will you calculate it? So, 2C, everyone answer 2C. Okay, B, A, B, C, D. Children, don't play MCQ game with P. You have to tell me the answer. Right, 2A, B, C, D, whatever is the answer, you have to tell the answer. So, look over here. So, together if I take all of them, it should be 100. For 100 divided by 4 is equals to what? 25. So 25% of them will be capital T. Okay, this also 25%, this also 25%, this also 25%, my dear children. So 25% plus 25% is equals to what? 50%. Your answer will be B. So those of you who got B as the correct answer, big kudos to you. And my dear children, please keep on answering, okay? Aye, Captain. Yes. Keep on answering. It's a really good thing, everyone, when they answer. Hi, Benetta. Okay. Children who are getting things wrong, please make it correct. I hope you got it correct. This is B. The answer is B. Going ahead with the next one, that is, which one of the following assertion about alpha thalassemia is incorrect? Can you tell me the answer for this? What is incorrect? My sound is very low. Can others please check? Is my sound okay or is it too low? Okay, 2B. Okay, we are in 2B question. Children, 3. We have reached third question. Hi Sudeep. Hi RJ. Third question, anyone answer? You are asked about what is incorrect in alpha thalassemia. Involves the HbA1 and HbA2 genes. I think it's correct, no? It's correct, no? It's low. Okay. Okay. Is it fine now? 3A, 3D, 3C. Okay, so many people are saying A, B, C, D. Everyone is there saying all the options. So it involves HbA1 and HbA2 genes. This is correct. Okay. It is associated with the mutation or deletion of genes on chromosome number 11th. Alpha thalassemia. No, Baba. Alpha thalassemia is not associated with this. It is associated with chromosome number 16. So this is correct, but this is wrong. Now, leads to reduction in synthesis of globin. That is also correct. If it was alpha globin, it would have been more correct. So this is your incorrect statement. So those of you got B, kudos to you. Ma'am 3B, ma'am 3B. Okay, Sagar, Anya. I'm not able to read out all the names, but yes. Pradeep, hi. Okay. Children, great going, great going. I hope you're getting things correct. Always remember alpha thalassemia, it's chromosome number 11, and beta thalassemia, alpha thalassemia is chromosome number 16 and beta thalassemia is chromosome number 11. Okay? Okay, cool. Let's go ahead. Now, which three scientists independently rediscovered Mendel's work? So Mendel worked really hard, okay, and people tend to forget him. But later on, his works were rediscovered by three set of people. Who were the three scientists there? Sparsha, great going. Benita, great going. Aisha, great going. Children, keep on answering, okay? Even if you get it wrong, keep on answering. Let's rectify our mistake. So Ambika says, okay, yes, okay. Children. Fourth question. If you are not answering like this, I would not be able to understand. Please answer like this. Okay, 4D. Okay, let's start. So Sparkle, Shripad, hi, and Preeti, Samant, Pradeep. Oh my God, so many. Neil, Sparkle, Tanush. Okay, Pradeep. So all of you are saying D. Let's go with D then. Yes, it is D. Rice, Correns, and Shemak, my dear children. Please make sure you know the year as well. Okay, just go through the year. And I saw Anya said, ma'am, important question in one shot, ma'am. I will be doing a 30-minute revision video. In that, I'll be including all the important questions. And it will be released in the morning, so it will be easy for you to revise. Okay, let's go ahead. Great going, children. All of you, crazily good. Awesome. Okay, now, what is the possible blood group of an offspring when both parents have AB blood group? Both of them have AB blood group, my dear children. Papa, Amma, both of them have AB blood group. AB blood group means that is codominant. Both of them will be having both the alleles. Now over here, a gamete carrying A, gamete carrying B, gamete carrying A, gamete carrying B. If this and this fuses, you get AA. That means it is A blood group. Yeah. If this and this fuses, you get AB. That is, it is AB blood group. This and this, you get AB again AB blood group. This and this, BB. So it will be B blood group. So I can get A blood group, I can get AB blood group, or I can get B blood group. So what is the answer for this? Who said C? Who said C, Baba? Where is O? Here we are saying that both the parents are having AB blood group, not A and B blood group. So your answer is B. Your answer is B. Okay, not C. Correct answer is C. How can C be correct? Listen, both the parents have AB blood group. AB. There is no O over here. So you can never get O. If the parent was, one parent is having A blood group, another parent is having B blood group, then there are chances of O as well. But here both of them are AB blood group. I hope you know the answer. Your answer is B. Going ahead, please correct it. Which of the following is incorrect regarding ZW and ZZ type of sex determination? ZW and ZZ type of sex determination. Now I'll tell you, this is the only one that is showing female heterogamety. Females over here are producing two type of gametes. Everywhere else it's a male that produces two type of gametes. Don't do that guesswork, okay, Sparkle? All question B option correct. Don't do that guesswork. It can be wrong. I will show you now itself. Sagar, tell me the confusion. What is the confusion? Okay, everyone is saying 6B. Let's see. So which one of the following is incorrect regarding ZW, ZZ type of sex determination is that here the females are heterogametic. Females having ZW type of gametes. So they will produce either Z or W. They are not homogametic. So it occurs in birds, that is correct. Females are homogametic and males are heterogametic. Is that true? No, Baba. Males are homogametic over here. They are going to produce Z or Z, nothing else. They are going to produce same type of gametes. So they are homogametic and females are heterogametic. So the statement is wrong, and this is what you had to find out. Sparkle, again you are correct, yeah. So this is the one you have to find out. Females heterogametic, wrong. Total number of chromosomes in both male and female, that is the same in both male and female. Yes, obviously it will be same, right? So this is the one. Praju, okay, ma'am fast. Okay, just a second. Children, for others, is my voice okay? If my voice is okay, just give me thumbs up. Okay, you do not need to type and all, just give me thumbs up, because some children are saying my volume is 300%, which is not even possible. But can you just check once? Answer 7th question until then. Give me a thumbs up if my voice is okay. Thank you, Shamant. So there are 2 people who are saying it's okay. Okay, there are 3 people. My dear children, please, if it cannot be like, for some it is audible, for some it is not. If you can, please unplug your earphones once and try, okay? Chalo, let's try. So haplodiploid sex determination is found in your book, only one. Mani says it's okay, okay, wait. Children, please try, okay, try opening, because a lot of you are saying yes, so I think it's audible. Now, let me check by myself, okay. In your book, only one. It's audible. It's okay, okay, wait. Please check. Okay, so your answer is honeybees for this question. They have haplodiploid sex determination, and only them according to your NCERT. What happens in haplodiploid? If it is diploid, a cell has diploid number of chromosome, that is double the amount of chromosome, it will be a female. If it has only half the number of chromosome, it will be a male, my dear children. It will be what? Male. Okay, this is haplodiploid sex determination. Vedant and all, please check once, okay, Baba? Now, going ahead with the next question. Great, all of you who said D is the correct answer. Good going, children. Now, in XO type of sex determination, what will be the thing that is correct? Females produces 2 type of gametes. Males produce 2 different type of gametes. I told you, female produces 2 different type of gametes. Only happens in what? Birds. Okay. Now over here XO, and that is ZW, right? This is XO. Now this is wrong. Males produce 2 different type of gametes showing homogamety. They produce 2 different type of gametes, but they do not show homogamety. Then this is wrong. Females produce gametes with Y chromosome. No, that is not at all possible. Now, males produce 2 different type of gametes showing heterogamety, that is possible. So, in XO type of sex determination, which is found in what organisms? Can you tell? XO type of sex determination is found in what organism? The letter starts with I. So, over here, males produce either X chromosome, or they produce gamete that does not have any chromosome. So, there are 2 types of gametes. You get it? Prajwal, ma'am, you teach very well. I like the way you teach. I'm thankful. Thank you. So that's so kind of you for telling that. And all of you, my dear children, who are telling the answers, great going. Mani, I am surprised you're a CS student and you were here. Thank you. Okay. But yes, good luck for your CS exam as well. Insects, great. So I'll read out certain names: Gauri, Preeti, and then Pradeep, Shinchana. That's nice name. Vedant, and Neil, Riaz, everyone, my dear children. Shamant. All of you, good, it is insects. For example, grasshopper, they show this, okay? Going ahead with the next question. When is it possible for a woman to be colorblind? Listen, a woman can be. Color blindness is what? Sex-linked recessive disease. Sorry, not autosomal. It is sex-linked recessive disease. And for a woman to be colorblind, okay, both the chromosome has to be affected. If one chromosome is affected, the other chromosome says, I am here to over-dominate and you won't be colorblind. You will be just a carrier. So, if a female has to be colorblind, then you should be getting it from both the father and the mother. If you're getting it from the father, my dear children, the father will be like this. The father is definitely going to be colorblind because there is only one X chromosome and that has to be affected. Now, talking about the female, the partner that this person has, what about her? 9D, everyone says 9D. Chalo, let's see. D, the father is colorblind, okay, true, and the mother is a carrier. Can the mother be a carrier, my dear children? Can the mother be a carrier? Yes, because suppose anyhow, if this and this comes together, my dear children, what will happen? The female child will be colorblind. So the mother can be a carrier. Now what about C option? The father is colorblind and the mother has normal vision. The mother has normal vision. Now normal vision, my dear children, it can be like this or it can be like this also. It can be carrier, it can be like this. So suppose this is the one, then the person would not be affected. So let's take the safer option, which is D1. Okay. Ma'am, still I have to read many chapters, should I? Okay. For children who have doubts like that, let me tell you, for children who are halfway through your things, okay, read your NCERT, just give yourself 15 to 30 minutes for reading the NCERT, not beyond that, okay. And then you have the PDFs. Do you have the PDFs? I have given in the WhatsApp channel. Go read the PDFs. I do not want extra views. If you are someone who is stuck, you have not completed the whole syllabus, you can go read the PDF and just get an understanding. But obviously, I will recommend you to listen, because while listening, you understand things better than just reading, because you have a lot of things. Okay. Going ahead. Now, if both parents are unaffected carriers for thalassemia, both the parents are unaffected carriers for thalassemia, which is an autosomal recessive disorder, what are the chances of pregnancy resulting in an affected child? Affected child, my dear children. So, both, suppose this is affected, they are carriers, that means they have, but only one gene is affected, only one allele is affected basically. So, what are the chances of the pregnancy resulting in affected child, my dear children? Again, make a cross. HB, HBX, HB, HBX, and keep on guessing the answers as well. This and this fuses, you get HB, HB, okay. This and this fuses, you get HB, HBX, okay, that means just a carrier. This and this fuses, again HB and HBX, again just a carrier. This and this fuses, you get HBX and HBX, that means this person is diseased. How much percentage? How much percentage? Total should be 100. So all of them should be 25, 25. So only 25% of them will be affected. Your answer is not D. Your question says, what are the chances of pregnancy resulting in an affected child, my dear children? Affected child. Here you get things wrong. Listen. Here you are gonna get things wrong. Where can I get the PDF? Baba, it's there in the WhatsApp channel. The link is there in the description box to the WhatsApp channel. Your answer is 25%. Okay, I hope you're getting. Children, if you're getting, can you just give me some heart? Otherwise, if you have any doubt, please stop me then and there. I am ready to solve. Okay. 11th one. In the F2 generation of Mendelian dihybrid cross, the number of phenotypes and genotypes are. Now, usually we know the number of phenotypes. In the dihybrid cross, you know, it is 9:2, 3:2, 3:1. 9:2, 3:2, 3:1. Where 9 of them will be showing the dominant trait, that is round and yellow. One of them will be showing the recessive parental trait, that is what? Wrinkled and green. Wrinkled and green. Now 3 and 3, you have to make new combination. So it will be round was with yellow, now this time it will be round with green. And 3 again will be wrinkled was with green, but this time it will be with yellow. So how many different phenotypes can you observe? Phenotypes as in physical appearance. One is round and yellow, the other one is round and green, the other one is basically wrinkled and yellow, and this one is wrinkled and green. So 4 phenotypes I can see. 4 phenotypes I can see. Okay. Nagaraj Pawar, Baba, you can ask the doubt. And hi, good morning. So 4 phenotypes. So I'll rule out this option because it is phenotype is 9. Now what about genotypes? Genotypes you have to remember. Genotypes mean genetic makeup. So round yellow can be like this, my dear children. It can be round yellow. This can also be round and yellow. This can also be round and yellow. So there can be multiple genotypes. So here I have put down. So these are the genotypes. 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 genotypes are there and phenotypes are 4 for this particular answer. Great. WhatsApp channel link in the description box. Okay. Great, my dear champions. Great. So great of you. Okay, Sparsha, Gauri, Deepak, everyone. Okay, now 12th. 37.2% recombinant Drosophila progeny obtained in the T.H. Morgan's dihybrid cross experiment with the phenotypes red eye color and normal body and white eye color and miniature body is due to what? So basically this is just to confuse you. All this information is just to confuse you. What is important? The recombinant percentage, that is 37.2. You know what? The highest amount of recombination is 50%. So 37.2, that means it's a good amount of recombination. Please understand, you two are best friends. If you two are not close enough, what will happen? You can get another best friend. So it's just like that. If the genes are linked to each other, my dear children, very closely, they will be showing less recombination. You and your best friend too close, that means less chances of new combinations to be formed. Okay, so that means closely linked genes is less recombination. Talking about loosely linked genes, that means they are far away from each other, they can show high recombination. So over here 37.2% is high recombination. That means the genes are loosely packed or closely linked. You tell me, the genes are loosely packed or closely linked. What will be the answer for this? It will be loosely packed, right? It will be loosely packed. So your answer is loosely linked genes and longer distance between the genes. This becomes your answer. C, exactly, all of you correct. M.S. Devadar, I'm so sorry. Some of you are saying to go fast, some of you are saying to go slow, which is why I'm in a confusion. I have time, but you do not have time. So yes, C is the answer. I hope you got it. Please not forget this. If the question was 1.7% recombination, what would have been the answer? Can you tell me the answer as well? If in the question it was said 1.7% of recombination, will it be closely linked or loosely linked? 1.7% of recombination means it is very less amount of recombination. So it is closely linked or loosely linked. Children, you have to answer this question. It is equally important to the MCQ given over here. MCQs can be reframed. MCQs can be reframed. Nagaraj, ma'am, I have studied only 3 model questions. Is okay. You have studied 3 model question papers. That is really good thing. Let's be happy for that. Now, my dear child, you can watch at least 2 marker, 3 marker and 5 marker video. Okay, 7 hours probably, or probably 6 hours. You will get a rough idea of the concepts as well. It will be really important for you, and then you can read down the question bank questions, which are the PDF is given in the description box. Okay, sorry, the PDF for the WhatsApp channel. The link for the WhatsApp channel is given in the description box. You can click on that, you'll get the PDF. Now it would be B, closely linked. Exactly. If it was 1.7%, it would have been the opposite. Great. Who all said the answer? Let me see. Neil D'Souza, good. Samanth, good. Jenica, all of you said B. Anusya, Samayaman, Jenica. Okay, great. Going ahead, let's go ahead. A pedigree chart is given below. Identify the trait responsible for this inheritance pattern. If without my help, if you are saying, okay, for this particular answer, C is the correct. I gave an alternative question. If it was 1.7%, what would have been the answer? Then it would have been B. 1.7% means there is recombination amount is very less. That means they are closely packed, okay, or tightly linked. Okay. Now, tell me the answer for this. Children, please put the number of the question, please. Please, otherwise I would not be able to understand. I'll say this is the correct answer and you will get it wrong. Get it? A pedigree chart is given below. Identify the trait responsible for this inheritance pattern. B, you're saying B, autosomal recessive trait. Why B? The answer is B, my dear children. Why B? Listen, whenever the recessive disease comes, okay, whenever the recessive diseases comes, no, just understand that it will skip generation. It will skip generation. As in, here the parents are not affected, but the children are. Here also the parents are not affected, but the children are. It is a recessive trait, which is why it is skipping the generation. Now over here, the male and the female are equally getting affected, so it cannot be sex-linked recessive disorder. It is autosomal. It does not have anything to do with the sex, so it will be autosomal recessive trait, my dear children. Okay, works? Chalo. You're most welcome, Nagraj. All of you who got 13B as correct, Samyama, Sinchana, Sparsha, Anisya. I'm so sorry if you're reading, if I'm reading out your name wrong, but yes. Deepak, Askar, Riyaz. Chalo, solve this question also. Question number 14th. In a dihybrid cross in pea plants, Mendel got 9:3:3:1 phenotypic ratio. It denotes that. My dear children, the last questions that I showed you over here, all of them basically are from the model question paper, so make sure you're not missing it out. Okay, Anu, set. And please, if you have your friends who are also preparing for the MCQ, you can ask them to join as well. Okay, chalo, 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 chalo. In a dihybrid cross in pea plants, Mendel got this ratio. It denotes what? 14th D. Gauri said 14th D. Riaz also said D. Okay, answer is D. Sanvi, hi, Sri Hari D. Okay, my dear children, when you say 9:3:3:1, you started off with round and, listen to me carefully, okay, I'm not writing. Round and yellow and wrinkled and green. You started off with that. If there was no segregation happening, if the chromosomes were not segregating, my dear children, your progeny, all of them would have been round and yellow and wrinkled and green. But it's not like that. Basically, law of segregation, or basically independent assortment, says that this chromosome can go with this chromosome. From a pair of chromosome, only one can go, right? Only one can go. So this can go with this, or this can go with this also. Either way, and that is why we have new combination. They are assorting independently. They do not have any dependence. So alleles of two genes are segregating. Alleles of two genes are segregating independently. That is why you get 9:3:3:1 ratio. These are the new types of things that we see. Okay. Okay, 14D, let's go ahead. Children, are we putting the same type of effects? And also one more thing, if you are someone who is preparing for KCET, okay, then we have Abhyas KCET test series and Lakshya KCET crash course. This one, you have to come to Bangalore and study with the teachers that we have. It's a 21-day course. If you want to enroll, you can call to this number. If you're not able to contact them, tell us, okay, in the comment section. Now this is the test series which we have got for ₹1499. So if you're interested, then definitely go for it. The link is in the description box. Okay, KCET preparation tips, I'll tell you. If I tell now, no, everyone is going to do dushan to me. So please, okay. Anu, yes, I'll say my sessions would be very much enough, but you have to spend some time. Yes. Vijay Lakshmi, thank you. Okay, Sparsha, you work. Thank you. Chalo. I'll thank all of you, but you have to do this. We'll start off with molecular basis of inheritance. Okay? First question. You solve this and I'll thank everyone. Okay. Shizuka, I will definitely tell you, okay, towards the end, or probably I'll write it down and tell you, or probably make a reel for you, but not now. Ma'am, drink some water. Oh, that's so considerate of you. Thank you so much, my dear children. I'll drink some water. Okay, after 3 questions, I'll drink some water, and at that time, I'll give you a break also. Diksha Karnataka and Vedantu Karnataka is not the same branch. Okay, Diksha is a entity that has tie-up with Vedantu, so it will be 2 different things. But yes, we are partners. Okay. So Dexter, Anu, and Riddhi, and everyone, Benita, Radha. 1B, okay, great. Lot of you are saying 1B. Let's see if it's 1B or not. So the 2 nucleotides of DNA are linked through 3 and 5 carbon of pentose sugar by what bond? What bond? So B, everyone says B. Yes, the answer is B. For those of you who do not understand, I would have fallen now. Sorry, just a second. So over here, 1, 2, 3, 4, and this is the 5th carbon. Okay, now 1, 2, 3, 4, 5, and this is the 5th carbon. Over here, the 5th carbon is attached to a phosphate group. The 3rd carbon is also attached to the phosphate group. This bond is known as a phosphoester bond. This is also known as phosphoester bond. You are asked between 3 and 5th carbon, so you have to include both these bonds. So it becomes phosphodiester bond. So B becomes the answer. All of you got it correct. It was actually gonna fall down. Oh my God. Yes. Next question. In biochemical categorization of transforming principle, which of the following enzymes affect transformation? First thing first, tell me the name of the experiment. The transformation principle experiment. What do you call it? Who put it down? Who put it down? Nishant, hi my dear child. You said B. You were absolutely correct. Okay. 2A, think, rethink, please rethink. You are asked which one of them will affect the transformation. So we studied that basically you have 2 strains. One is the dead S strain, and the one is live R strain. And then when we put it in the mice still dies. How? Because something was transforming the R strain over here. the live R strain over here. What was this something that was transforming? It was DNA. And how did they come to know about that? Basically, Avery, MacLeod and McCarthy, the three other people after the Griffith, they came and they used DNAs. When DNAs was used, DNA was getting damaged. And when DNA was getting damaged, do you think transformation can happen? No. So which of the following affects the transformation? It will be DNAs. Okay. Benita, Sparsha, Leela, hi, exam expert. Yes, it is Griffith's transforming principle. Kudos to you, Samyama. Okay. Leela, all of you, great, great. None of this, Sahil. Okay. My dear children, you are absolutely great. Going ahead with the third one. Okay. The experiment that proves the semi-conservative principle of DNA replication. Now, this is an easy question. Can we get the answer for this? Endless warrior, thank you. Okay, the experiment that proves the semi-conservative principle of DNA replication is what? Very much important. Please, please go through this experiment if you have all the experiments. Griffith experiment, okay, Avery, MacLeod, McCarthy, and then what? Hershey and Chase experiment, and then this experiment also. 3D. Okay, Gauri, Preeti, Gauri again, okay, Leela, Riddhi, Benita, Deepak, Riddhi, set C, check, 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 check. It's D, okay. Answer is D. It is Michelson and Stahl experiment. What was this experiment conducted on? It was conducted on E. coli, my dear children, where radioisotopes were used. Different radioisotopes of what? Nitrogen. One was 15 and the other one was 14. Please go to the experiment. Very, very important experiment. Okay. There is nothing like more trustable. We are trustable. You can follow us. Okay. Now, it depends upon children to children whom they are comfortable with, right? So, yes, obviously, everyone is doing their best. So, you can follow what is the best for you. Okay. Now, fourth, RNA polymerase 1 transcribes what? Very important question. We have 3 RNA polymerases. In the case of eukaryotes, we have 3 RNA polymerases. That is RNA polymerase 1, 2 and 3. You should be knowing what RNA polymerase, what do? They transcribe. They make RNA out of a DNA, right? Now, each of them have different functions. You should be knowing the answer for each of them. So, 4A said 4A. Gauri said 4A. What about others? Yes, Srihari, it was 14 nitrogen and 50 nitrogen. Great. And Deepak 14C, 4C. Okay. ASG says D, Sahil says A. Okay, so it's like more of an MCQ for me, right? The answer for this is, always remember, RNA polymerase. Okay, RNA polymerase 1. So it's primary. 1 means its primary priority is to make rRNA. That is ribosomal RNA. Ribosomal RNA, that is rRNA. Now, this question can come on the day of exam as well to confuse you. So here I have written all the things. So RNA polymerase 1 transcribes rRNA. Okay, that is the first priority. Now, RNA polymerase 2 transcribes precursor of mRNA, the heterogeneous RNA, that is hnRNA. That is the second priority. Now, the third priority is basically what? It is to make tRNA, it is to make 5sRNA and snRNA, small nuclear RNAs. So, you have to remember like that. If you remember both these things, you know what does not fit in the category. And that exception would be this one. Okay? Yes, please, Nagraj. Okay, so your RNA polymerase 1 transcribes rRNA, that is the first priority, ribosomal RNA, 20 is S subunit, 18 S Swedberg coefficient, sorry, and 5.8 Swedberg's coefficient. Now, RNA polymerase 2, it is going to code hnRNA, so this becomes your answer. Remember this, you will be able to guess the answer. Easy trick. Okay, so all of you who said 14A, kudos to you. All of you who didn't say, it's okay. Let's try more, and this is how we study. Now, GGU, GGC, GGA and GGG all code for glycine, my dear children. Which salient feature of the genetic code does it prove? What is the proof? Which is the genetic code feature that it proves, my dear children? All of them are coding for the same amino acid. So, this type of feature is known as what? Leela, superb. Thank you. 5B. 5B. Everyone says B. Shall I log B? Shall I log B? Yes, it is B. It is B. So, this feature you can remember by degeneracy. So, glycine things, sorry. So, the glycine things basically, no, let's not take a story over here. So, it's DGU. When you have multiple codons for single amino acid, it is known as degenerate codons. Now, when I say that GGU is coding for glycine, if just this is mentioned, what do you understand from this? That it is unambiguous. GJU does not code for anything else but just glycine. Okay, so it is faithful. You can remember like that. But this feature is known as degeneracy, where multiple codons are there for one particular amino acid. Okay, Nishant, hi. Okay, now children, are you enjoying? Are we studying? Do you think it's useful? Okay, aminoacylation of tRNA occurs during what? Aminoacylation of tRNA occurs during what? Very important question. Come on. Are you? 6B. Gauri says 6B. Leela says B. Riddhi says B. Everyone says B. Shall I log B? What about others? Where did everyone go? Shaukat Ali, hi. Benita, Sinchana, everyone says B. Answer is B actually. What do you mean by aminoacylation of tRNA? Your tRNA looks like this, my dear children. Okay, where it has a 5' end and a 3' end. Now onto this 3' end, when one amino acid comes and attach, this process is known as aminoacylation. This process is known as aminoacylation or charging of tRNA. Okay, and this is very important for the process of translation, because you have an mRNA. mRNA suppose has a codon AUG. So there will be an anticodon loop, my dear children, where it will be UAC. It will come and attach, and basically that means that this particular codon is coding for this particular amino acid, that is what? Methionine. So it's a very important thing, and it is there for translation. Okay? Sparsha, yes, you also said the answer. So I actually casually looked over here, which is why I could not. Okay, going ahead. What do you want the duration to be? So instead I can be worried about duration, or probably I can solve all the questions. You can choose. So we can stop if you want in the most high weightage chapters. Either way. Okay, we'll see. Now in a single stranded DNA, it's a very important question, please remember this. Yes, this is known as activation of tRNA as well. Now in a single stranded DNA, if the ratio of A and G is 60:40, then what is the ratio of T and A? Gauri says 7A, Riddhi says A. Children, do not talk about Insta ID over here. Tomorrow we have biology exam, please. Thank you so much, Nishant. Okay, children, be very careful while answering this question. I am telling you again and again. No one as of now has given me the correct answer. Okay. Okay, so the question says, in a single stranded DNA, there is only single strand. Here if A is to the power, what say, A is to G is 60:40. T can be any number, no? T can be any number. Even C can be any number. So can I just, with the help of A and G ratio, can I find T and C? No. If it was double stranded, then I know that wherever A is there, T will be there. So it will be 60:60. You get it? But here it is single stranded, so your answer becomes none of the above. So what is the answer for this? Is this a good question? Is this a good question? It is a good question. So your answer is D, none of the above. We never know. It is a single stranded DNA. Arif, hi. Ma'am, B is correct. How can B be correct? Baba, think logically. Again, it's single stranded. Okay, if single stranded. So wherever A is there, A is there. Wherever T is there, T will be there. It's not coding like this. It's not bonding like this. No, it bonds like this. You get it? So it does not matter. So your answer is, oh yes. What is the composition of catalyst involved in peptide bond formation? 28S, I suppose. Just check it once, okay? Okay, going ahead. So this was an eye-opening question, I suppose. It's okay, do not be sad. Now next question, again, this is also a very important, tricky question. So please use your brain. Ma'am, can you explain why D is correct for 7th one, please? Yes, Gauri, I can. So listen, here it is mentioned, in a single stranded DNA. So if a single strand DNA, if A ratio power G is basically 60 and 40, okay, over here suppose A is this many. Just because A is this many, can I say T is also this many? No, because I can identify the number of T if it was bonding like this. But this is a single stranded thing, so A can be as much ever as it wants and T can be as much ever it wants, because it is not bonding over here. You get it? If you are asked about double stranded, then things would have been different. Gauri, I hope you understood that. Now, in a mRNA, this undergoes translation. How many types of amino acids are present? D. Okay. Sinchana also says D. Gauri, okay, thank you. Okay, welcome. If you still didn't get it, you can tell me in the comments. Okay, I'll explain it properly while writing. So, my dear children, you are wrong. You are wrong. Why? In which direction is mRNA read during translation? mRNA is read in 5' to 3' direction. What is the direction mentioned over here? It is 3' to 5'. So this is not a stop codon and this is not a start codon as well. So if you calculate it like that, it's way wrong. Okay, now let's start off with the actual deal. So AAU codes for one amino acid, UUU codes for phenylalanine, again UUU codes for phenylalanine, again UUU codes for phenylalanine. So what type of amino acid? It's only one type of amino acid, that is phenylalanine. So this is the second type of amino acid. Now GUA will also code for some amino acid, so it will be 3 different types of amino acid. How many types of amino acids is being asked? So 3 different types of amino acid that is being coded by this particular thing. So your answer becomes B. This is the 8th question. Yes, Parsha, please. So your answer becomes B. Now I hope you understand, because you do not have to read from 3' to 5' direction, you have to read from 5' to 3' direction. So AAU 1, UUU, always UUU codes for phenylalanine only. So I cannot take it as different type of amino acids, I'll take just one type, phenylalanine. And then GUA, again another type. Okay, first Heli Baba, okay, I'll go. Children, I have to make others understand also. So B is the answer. Let's go ahead with the next one. I hope everyone got it. Please do not be sad. Now, which of the things these are correct? Tell me, which of these are correct? All the children who were there, I understood. Thank you, Sparsha. Thank you for telling. Okay. Ma'am, UA is a stop codon. Nishant, UAA, not in 5' to 3' direction. You have to read it like this, not like this. You're getting things wrong. mRNA is read from 5' to 3' direction. Okay, now tell me the answer for this. Which of these are correct? UTR regions, untranslated regions, are located on the DNA. Okay, UTR is present both at 5' before the start codon and also at the 3' after the stop codon. UTR provides what? Provides stability to mRNA. UTR is required for efficient translation. These 3 things are correct. UTR, untranslated regions, are not present on DNA, it's present on mRNA, okay, before the 5' and after the 3'. Okay, here it will help in initiation, there it will help in termination, and it also provides stability. It is required for efficient translation as well. So this becomes wrong. So 2, 3 and 4 is the correct one. 2, 3 and 4 is there over here. So D becomes the correct answer. D becomes the correct answer. Your question is what is correct, not incorrect. 9 children, 9D. Wahida, Abhi, I hope all of you are answering some different question, not this. If you are answering 9th question, then it is about correct, and these 3 are correct ones. So this becomes the answer. There is no untranslated regions on the DNA. Got it? I'm so sorry. My name is Jasmine Joy. I'm so sorry for children if I'm going wrong. Some are telling me to go fast. Not wrong, I mean fast. Okay. Even my head is gone. Okay, methyl guanosine triphosphate is added at the 5' end of hnRNA in a process of what? So I hope you know that when you say there is DNA, and this DNA, when it is transcribed, it is not transcribed directly to mRNA. It is actually transcribed to something that is known as hnRNA, and it will have all the information that is there in the DNA as well. It will have exons that codes for amino acid. It will have introns that does not code for anything and everything. So hnRNA basically has to undergo processing. Okay, it has different type of processing. One processing is where you remove the introns. This is known as splicing. Then other processing is known as capping. Your answers are correct. Gauri, Sinchana, Sparsha, Saurabh, Wahida, Riddhi, all of you, my dear children, starting from, yeah, Wahida, Riddhi, Saurabh, every one of you, ASG, correct. This is known as capping, where you are adding what? Methyl guanosine triphosphate. Why do you add it? Because this is there in the nucleus. From the nucleus, when it goes outside, it does not have to get cut by any enzymes. So to safeguard it, you have capping. Apart from that, there is tailing also, where you add adenine group. Okay, there is adenine group as well. So your answer is B. All of you got B. Kudos to all of you. Now going ahead with the next thing. Human insulin is produced in E. coli cells by rDNA technology. This proves that genetic code is very easy question. Okay, human insulin, it is produced in E. coli, my dear children. This proves what feature about genetic code? What feature about genetic code? A, universal, B, triplet codon, C, degenerate, D, initiator codon. Samanth, yes, 10th answer was B, Baba. All of you who gave answer B for 10th, it's correct. Now we are dealing with 11th question. Tell me 11th, A, B, C, D, what is the answer? Saurabh said A, Gauri said A, Mahateb. Baba, can you just check your answer once? Binita said A, and then Safura A, Shaukat Ali A, Sinchana A, Sparsha A, Leela A. Okay, all of you A, yes. Listen, E. coli is able to produce our insulin. That means our genetic code is what? Universal. It is the same in E. coli and same in us. So it is universal. There are only few exception of genetic code which is not universal. That is mitochondrial DNA. It is different from ours. Okay, apart from that, there are some protozoans who have some different set of thing. Okay, rest everything is same. I hope you got the answer. Abhi Kumar, Baba, all of you. Vinay also, great going. Now, next question. If A and B DNA are of two mothers, okay? Now C is the DNA of the child. C is the DNA of the child. Based on the banding pattern of DNA fingerprinting, what can be drawn from this thing? What can be understood? Just check through. Okay, if you want me to zoom in, I will zoom in a bit. It looks like this. This is C, this is A, this is B. These two are mothers. There is a child. Whose child is it? Direct question. Whose child is it? Tell me, answer 12, A, B, C, D. What is the answer? Gauri said A. Gauri so fast. Okay, great. Gauri said A. Are you sure it's A, Baba? Are you sure it's A? Can you just check once? Banding pattern. See for the banding pattern, my dear children. Over here, this banding pattern and this banding pattern exactly similar. That is of B and C. So your answer becomes what? Child C belongs to mother B. Child C belongs to what? Mother B, my dear children. So your answer is B. 12th, B. Okay, please look for that. And if you got it wrong, please do not worry. That's how we learn. Okay, next, very important question again. Okay, match the following. Alec Jeffrey, F. Sanger, Jacob Monod and Griffith. So you have this and this. You have to match. So who gave DNA fingerprinting? Who gave DNA fingerprinting, my dear children? It was Alec Jeffrey who put down DNA fingerprinting. Who spoke about annotated DNA sequencing? It was F. Sanger who did that. So A goes with 4. A goes with 4. You have two options that A goes with 4. Let's eliminate these two options. Now let's talk about B. F. Sanger is with 3. F. Sanger, B is with 3. So there is only one option saying that. Now C with 1. Is C with 1? Yes. Jacob and Monod gave lac operon and Griffith gave the transforming principle. So B becomes your answer. 13B. All of you got it correct. Kudos to all of you. Okay? Children, make sure you are not getting demotivated when you are getting answers wrong. This is the best way to learn things. Okay? So please be happy. So here I have marked the answer wrong. Do not consider it. This is the correct. Correct. Okay, statement. Chalo. 14th. Now tell me this is easy or not. Statement 1 says deoxyribonucleotide triphosphates, DNTPs, acts as substrate during DNA replication. As in, when I say DNA is replicated, I have one strand and another strand. The substrate, for example, I have A, T, G, C. So here again you have T. These are the substrates, right? So these are deoxynucleotide triphosphates. These are deoxynucleotide triphosphates. So, are they the substrate during DNA replication? Yes. Statement 2 says, deoxyribonucleotide triphosphate also acts as source of energy in the process of DNA replication. Is this correct? Is this correct or not? 14th A, someone says 14th A, okay. Both are correct. Think. Everyone says is correct. So my dear children, when you talk about a nucleotide, 1, 2, 3, 4 and 5. This is the pentose sugar. On the pentose sugar, you know, 5th carbon we have phosphate group. You know what? Actually, the phosphate group comes in triphosphite form. 3 phosphates comes to attach. But what happens? Only one attaches. There is a bond break over here. There is a bond break over here. And when this particular bond breaks, that's when you get energy to form another bond with another pentose sugar. So this is also true. So I'll say all of you who got it correct, kudos to you, have read your NCERT really well. So your answer is A. And those of you who didn't get it correct, don't think you didn't read it properly. It happens, okay? We miss things. Okay? Tell Erwin Chargaff's rule. So Chargaff said that basically the number of pyrimidines, okay, sorry, number of purines is equals to what? Number of pyrimidines. A plus G is equals to what? T plus C as well. Okay, because you know A pairs with T and G with C in a double-stranded DNA. My dear children. Okay, going ahead. Okay, next. In pea plants, each character is governed by a gene which is, again tell me the answer. 15th. In pea plants, each character is governed by, hi Gulsita. In pea plants, each character is governed by genes which is what? Monocystronic, polycystronic, operon, intron. Yes, purines is equals to pyrimidines, as in A plus G is equals to C plus T plus C. Okay, that is the thing. Gauri, I can actually do this. If I do it now, it will be a problem. Can you just please check the video? I can put the timing of the video later on. Can you? Is it fine? In the comment section. Okay, where I have explained measles and install. 15A, everyone says A. Yes, your answer is A, my dear children, because eukaryotes have monocystronic genes. That means in one particular gene, okay, in one particular thing, okay, you will be having just one gene coding for one particular codon. In a transcription unit, you just have one structural gene, each having a promoter and terminator for its own. So this gene codes for one particular protein. This happens in the case of eukaryotes. In the case of prokaryotes like bacteria, what happens? You have multiple genes, gene A, gene B, gene C, each together have one promoter and one terminator. For example, lac operon, you have Z, Y, A, 3 genes coding for 3 different things. So that is polycystone. So pea plant is a eukaryote, plants are eukaryotes, so the answer is A. Okay, going ahead with D. Okay, Sanjay. Over here, the number of nucleotide pairs present in the DNA of the primary oocyte of a newborn is, again be very careful. Be very careful while you answer this particular question. Ma'am Fan Hakuli, I hope you are saying that. I don't know, I should not be saying that. Okay, anyways, yes, children, I am in the phase of learning Kannada, so please don't feel bad if I am not able to speak it properly. Yes, the number of nucleotide pairs present in the DNA of the primary oocyte. Primary oocyte is in which stage? It has diploid number of chromosome. Okay, it has just started undergoing meiosis, it has not completed. When it will complete meiosis, that's when you will call this as what? Secondary oocyte. This is what? The polar body. This is secondary oocyte, this will have half the number of chromosomes. So, in general, 2n, that means 6.6 into 10 to the power 9. If just n, then that means 3.3 into 10 to the power 9. Okay, so your answer is what? Answer is what? B. 16B is the answer. Dimple, you said that I will study very fast, you will pray for me, right? Something like that. Okay. So, yes, B. Yes, you all are correct. Let's go ahead with the next question. Children, I hope I am helping you. I am not going really fast. If I am going fast, I am so sorry. Akshita, that's so kind of you. So, in lac operon, if mutation occurs in the Z gene, what do you mean by that? What will happen if the mutation is occurring in the Z gene? Sparsha, I am from Kerala, but I was born and brought up in Madhya Pradesh, Indore. Okay, but I am a native of Kerala. And now I am staying in Kerala as well, but I'm working in Bangalore. So yes. Children, in lac operon you have Z, Y and A gene, each coding for each things. I am asking, if Z is affected, what is the thing that is not going to be formed? Sakib, primary oocyte is not haploid, it is diploid. We usually make that mistake. Please do not make that mistake. It has just started undergoing meiosis. When it completes meiosis 1, that's when it becomes haploid. So secondary oocyte is haploid, not primary. Okay. Your answer is B, pakka B, because yes, Z codes for beta galactosidase, Y codes for permease enzyme, and A codes for transacetylase. Zia. Okay, you can remember like that. So your answer is this. If this is mutated, beta galactosidase will not be synthesized. Okay. Going ahead with the next one. All of you getting your answers correct. Kudos to you. Now, a DNA segment has a total. Okay, again, a tricky question. Please be very careful with this question. Okay, a DNA segment has a total of 1000 nucleotides. Okay, 1000 nucleotides, out of which 240 of them is adenine. Is what? Adenine. How many pyrimidines bases this DNA segment possesses? We have two types of things, purines and pyrimidines, right? Purines is A and G. Pyrimidine is T and C. In the case of R and A, you have U instead of T. Okay. Now you are asked, if 240 adenine is there, how many pyrimidine is there? How many pyrimidine is there? How do you find the answer? You are saying A is the answer. A, B, C. So you are giving me options, all of you. Saurabh says D is the answer. Let's see. So my dear children, if A is 240, T will also be is equals to 240 itself. Now 240 plus 240 is equals to what? 480. So A and T together it's 480. How many total nucleotides do we have? We have 1000. We have to minus this. When you minus this, what do you get? You get around 1000 minus 480 is equals to what? 520. Right, 520. Now 520 you have to divide between two things. What are the two things? One is G and the other one is C. So if G can be 260 and C also will be 260, then the total thing, if you now calculate 240 plus 240 plus 260 plus 260, it will be 1000. So your answer is what? Answer is what? Are you? Just a second. My dear children, it is not 500, it is 520, okay? It is 520, not 500. So your answer is B. Your answer is B. Please see my answer. Where are you, Leela? 18th B. Yes, exactly. Your answer is 520, not 580. I hope you got it correct. You are asked the number of pyrimidines. Wait, just a second. 1000. Yeah, so basically 240 plus 260, it is 500 only, not 520, it is 500 only. Okay, not understood? You didn't understand? I'll tell you. Listen to me carefully, okay? Children, please slow down. It's for them who didn't understand. It's a very important question. So we have 1000 nucleotides. Okay, in that you are saying that adenine is 240. Okay, you are asked to find the number of pyrimidines. Pyrimidines are T and C. Purines are A and G. Since we know A, we can find T. They bond with each other, so it is also 240. So out of 1000, 240 plus 240, that is 480 is A and T. Okay, is A and T. Now going ahead, we want to find what? G and C ratio. So 1000 minus 480 is equals to 520. Now 520, if we half it, we can find that G is what? 520 divided by 2 is basically what? You get 260 here, and C is also what? 260. Agreed? You got this? Now you are asked the number of pyrimidines. So you have to calculate 240, that is thymine, and 260. When you calculate it, what do you get? You get 500. So this becomes your answer. Okay? I hope you got it now. Chalo, going ahead, children, I suppose you got it. Please give me a thumbs up if you got it. If you didn't, tell me now only you didn't. Okay? Okay, we are going ahead with the next chapter, that is sexual reproduction in flowering plants. But before that, every one of you, please tell me if you got it or not. Yes. So, time for drinking some water. What is transgenic animals? Animals which have their genes altered. They are transgenic animals, genetically modified animals. Children, please drink some water. Hydration is very important, especially in biology, because we are biology students, right? Nishant, it is 500, okay, it was my mistake. When you calculate T and C, it is 500. Ma'am, please say I'm okay. Okay. Going ahead with the next thing. Ma'am, what is splicing? Splicing means when you're removing introns. You know, introns are there which do not code for anything, and exons are there which codes for the amino acids. So when you remove the introns, this process is known as splicing. Yes, stay hydrated you all. Okay, children, are we ready for the next thing? I'm in fasting, Ayyo. Please take care of yourself. Yeah, dioecious plants are, question number 1 of sexual reproduction in flowering plants. Dioecious plants are what? Autogamous, both autogamous and gynogamous, both gynogamous and xenogamous only. Artistic soul, please take care of yourself, okay? Everyone says B, everyone says B. Let's see. What do you mean by dioecious plants, my dear children? Dioecious plants means di as in 2. You have 2 different plants, each having one sex. So this is one plant with the male flowers, this is the one that with female flowers. So over here, definitely what will happen, the pollen grains can travel from here to here only. That's the only way of pollination, and this type of pollination is known as xenogamy. So your answer becomes D. Your answer becomes D. Manikanta, it's not exons are added and introns are removed. Listen, this is H and RNA, heterogeneous nuclear RNA. It has exons and introns. Exons and introns. You remove the introns, this process is known as splicing. And when you join the rest of the exons, you get the mature mRNA. You get the mature mRNA. Splicing means just removal of introns, okay, from the H and RNA. Geeta, on what basis? Okay, these are the questions from the question bank and also from the model question paper. So we can expect a mix of this coming, right? So yes. Ma'am, please tell I didn't read anything. First thing, you should have read something, and just a second, okay children, I hope you got this answer. This is xenogamous, this is only xenogamy, that is the only thing possible, so D becomes your answer. If it was other type of plants, it would have been given different answer. Okay, going ahead to the next question and answering one of your questions. For then, until then you can solve all this thing. So, ma'am, please tell I didn't read anything. What you can do is, please start watching all 1 marker, 5 marker, 2 marker, 3 videos, at least that. And I have put the PDF for the top 80 plus question, that is the questions from the question banks. So you can just read through the PDF at least as of now. Just read through the PDF, but listen to me in the 2 marker, 3 marker and 5 marker, and also the 1 marker. And 1 marker you're also already listening, but yes. Okay, it's very much important. Now, pollination takes place between 2. Yes. Sparsha, ma'am, which chapter we can skip? I would not recommend skipping any chapter if you are scoring, if you want to score out of out. But yes, if that's not the case, then biodiversity, but you should not be skipping it. You know, it's scoring. Chotu chapter, if you just solve, okay, biodiversity and conservation, if you do not read NCERT, just go through questions, it will be really easy for you to solve. I'll take 1 chapter down. Okay, 2B, everyone says B. Endosperm persists in the mature seed in groundnut and beans. Listen, my dear children. So in some seed you can break them down to 2. They are known as dicots. What happens in dicots? They have 2 cotyledons. How do they form cotyledons? They form the cotyledons with the help of endosperm. So they do not have anything left. Okay, so they are known as non-albuminous. They are known as non-albuminous because they have taken all the endosperm to form the two cotyledons. So basically your groundnut, your beans are dicots. They are dicots. There the endosperm does not persist. But in the other group of organisms, that is monocots, my dear children, what will happen? This endosperm, some amount of it persists because only one cotyledon is formed. Instead of having two cotyledon, they have only one cotyledon being formed, which is why endosperm persists. So wherever you see monocots, they are the ones that have endosperm persisting. Castor and coconuts are monocot. Pea and castor, pea is a dicot. Beans and pea both are dicots. Groundnut and beans both are dicots. So how do you remember it? Dicots you can break into two. So pea and all you know, beans and all you know. You can imagine if you can break into two, that means it is dicot. They would not have endosperm. Okay. Pearl says coconut has free nuclear endosperm, which is tender coconut water. Yes, and cellular endosperm, which is the kite kernel. Exactly, great, very important question. Okay. Going ahead with the next question, question number 3. Question number 3, that is in statement 1, in Viola, both autogamy and geitonogamy is prevented. In Viola, both autogamy and geitonogamy is prevented, and statement 2 says geitonogamy is similar to autogamy since pollen grains comes from the same plant. Tell me the answer for this. Children, I will be coming to your questions, okay? I will be coming to your questions. Which to skip? I want above 50. That's what I said. I would not recommend Sparsha. I cannot just say to skip things, but yes, as I say, if you do not want to read the NCERT, at least go to the question ones of biodiversity and conservation, organism and population, and ecosystem. The last chapters. Just read the questions at least. Okay. So everyone is saying C, one is wrong and 2 is correct. Okay. Let me see. 1 is wrong. So in Viola, both autogamy and geitonogamy is prevented. It is wrong. Why? Because Viola has 2 types of plants. One is clistogamous, where what is happening? The female and the male part is there. Okay. From here to here, anyway pollination will be happening. Anyway pollination will be happening. This type of pollination is known as autogamy. In the same flower when it is happening, it is known as autogamy. Now they have chasmogamous flowers, that means open flowers having male and the female parts. Here also they will be having. So from here to here, if pollen grains are going within the same plant but between 2 flowers of the same plant, geitonogamy is also happening. Geitonogamy is also happening. These are known as chasmogamous flowers, the open flowers. These are clistogamous flowers. So autogamy is happening over here, geitonogamy is happening over here. So both of them are happening, but it is written prevented over here. So this is wrong. Now second statement, geitonogamy is similar to autogamy. Since the pollen grains come from the same plant. Yes, it is similar. It is not written as same as. Okay, it is similar. Okay. To autogamy because pollen grains are actually coming from the same plant, right? So I will say both A and, sorry, so statement 1 is basically false and statement 2 is correct. So D is the answer. Who all got D? Oh my God, so many of you. Designer Shanti, are you? Okay, I see repetition. Okay. Leela, Shaukat Ali, all of you said D. My voice is not audible. Answer is D, okay? This is wrong. Please go through it once. Going ahead with the next question. Select the incorrect statement with respect to vegetative cell of pollen grain. Children, I hope my voice is audible. Designer, Shanti, can you just check Baba once? And please tell the answer in 4 A, B, C, D. I won't understand what answers are you telling, okay, if you just write the alphabets. You have to find the incorrect statement regarding the vegetative cell of the pollen grain. So if you talk about the pollen grain, my dear children, you have 2 types of cell. One is vegetative cell, which is the bigger one, and in the cytoplasm of the vegetative cell lies the generative cell. Generative cell. So here it has irregular nucleus, vegetative cell. This guy has a spindle shaped nucleus. Vegetative cell is therefore providing nourishment because this generative cell are the one that are going to break them into what? Gametes later on. Now, since I told you about this, can you tell the answer? It is bigger than generative cell. The vegetative cell, is it bigger than generative cell? It is correct. Oh, yes. Has abundant food reserve? Yes. Has a larger irregularly shaped nucleus? Yes. Floats in the cytoplasm of the generative cell? No. Generative cell floats in the cytoplasm of this, so D becomes the answer. 4D, 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 4D. Everyone who said D, you got it correct. I'm not reading out who got it correct, okay? Because otherwise I think others will kill me. Okay, but children, I'll read it later on, okay? I'll be going through all of you who are participating really, really well. I'm so happy. Now, Zuhra has C, okay. Now, which one of the following statement is not true? Is not true. Not true, my dear children. Pollen grains of many species cause severe allergy. It is true. Stored pollen in liquid nitrogen can be used in crop breeding program. It is also true. Tapetum helps in dehiscence of anther. Wrong. Tapetum does not help in dehiscence of anther. There is porogenous tissue, which are the cells which are going to form the microspores later on the pollen grains. For providing nourishment to this, you have the tapetum. Tapetum is just for nourishment. It has binucleate, two nucleus and all. Please, it is there for nourishment. So your answer is C. This is the incorrect statement. Exine of the pollen grain is made up of sporopollen. Very important question. Exine. Okay, so you've talked about pollen grains. It has plasma membrane, it has intine, it has exine, the irregular exine. Okay, and exine is made up of sporopollen, which is resistant to attack of alkali, acid, pH and everything. So that is also correct. Your answer is C. Going ahead with the next question. I am actually shivering. Guys, don't scare me by saying that please go fast. Some are saying go slow, some are saying go fast. Okay, from the statements given below, choose the option that is true for a typical female gametophyte of a flowering plant. What is true, my dear children? Okay, true, true, true. So it is 8 nucleate and 7 celled at maturity. Is it true? Yes, it is true. It is true. Why? We have 3 cells, 1, 2, 3, antipodal cells. Okay. Now 1, 2, 3. This is synergid. This is also synergid. This is egg cell. Okay. There are 2 nucleus. So if and this whole thing is a cell. So over here, how many cells are there? 1, 2, 3, 4, 5, 6, and this is 7. 7 cells are there, but nucleus 1, 2, 3, 4, 5, 6, 7, 8 nucleus is there. So it is 8 nucleate and 7 celled at maturity. This is correct. It is free nuclear during development. Is it free nuclear during development? Free nuclear during development, my dear children? Yes. So while developing an embryo sac, the nucleus divides, continuously divides. That is free nuclear condition. And then later all cell wall and everything forms. So this is also correct. Okay. Now it is situated out inside the integument but outside the nucleus. Wrong. Wrong. If you talk about the ovary, if you talk about the ovule, inside the ovule you have the nucleus. This is the nucleus. Now the ovule is covered by the integument, my dear children. You have 2 integuments. So integument is outside, nucleus is inside. It is situated inside the integument but outside the nucleus is wrong. It is situated inside the nucleus also. Here is the embryo sac located. Okay, so this is wrong. It has an egg apparatus situated at the chalazial end. Always remember, egg apparatus. That means you're talking about the egg cell and the synergids. They are never in the chalazial end. They are in the micropylar end. Why? Because it has a small pore. Through the small pore only the pollen tube will be coming. So your answer is what? A and B are the only correct options. So C is the answer for this. 6C. Okay. Got it? Children, if you're not getting it correct, it's fine. I'll ask you one thing to do, okay? Have a notebook, write the question number there. Come back, visit to it later on, okay? Works? Now, identify the correct sequence of arrangement of cells in the mature embryo sac from the micropylar end. From the micropylar end, what is the sequence? Please tell in the form of 7 A, B, C, D. Don't just give me random alphabets. I'm going to kill you. I won't kill you. Can you just tell me the answer for this 7th? Until now you guys were saying, ma'am fast, ma'am fast. Now when I am fast, you guys are not fast. Sanjay says A. Sparkle says A. What A? 7A? Yes, good. Good children. 7A. Binita says A. Again. B. Shaukat Ali. Okay. My dear children, the answer is A. Synergids, then egg cell, then the central cell, then the antipodals. I just explained over here. See, micropylar end to this. So first comes synergids. Actually, it does not look like this. Instead, it's like this. So there are 2 synergids and over here you have the egg cell. So synergids first, then the egg cell, then the central cell, and then the antipodals at the end, because micropylar end is being mentioned over here. Here it says from the micropylar end. So this becomes your answer. Synergids, then egg cell, then central cell, then antipodals. Okay? Works? Okay? Not C, not C. C is what? Synergid, central cell and then egg cell. No. Synergid and egg cell comes together always. That is forming the egg apparatus. Okay? Going ahead, Ammu and everyone, Zahra. 8th question. Even in the absence of pollinating agent, seed setting is assured in what? Even in the absence of pollinating agent, the seed setting is assured in what? Pakka seed will be formed. Anyhow, cucurbita, papaya, commelina, maize. It's a very straight, direct thing from your NCERT. My dear children, seed setting is always pakka in clistogamous flowers. Clisto, CL is there, that means closed flowers. Over here, they do not have an option. The pollen grains will be landing over here. And where do you find clistogamous flower? 3 plants. Viola, oxalis, camelina. Viola, oxalis, camelina. COV or VOC, either way, you remember. Video call, okay? Viola, oxalis, camelina. So your answer is this, C. So all of you who got C as correct thing, big kudos to you. My children, you are going really great. Okay? Next question. So I'm not even reading out your name because of everyone saying fast. So yes, select the correct sequence of anther wall layers. So you answer for this. Until then I'll say the names. So starting from Nirmala, Prateek, Deepak, Ammu, Vinita, Darshan, Safura, Srihari, Sanjay, Neel, Sinchana, Safura, Ammu, Shaukat Ali, and everyone getting Zohra, every one of you, great. Common pansy, yes, it is known as common pansy as well. Ammu, I told you. Okay, chalo, select the correct order of anther wall layers, my dear children. So you have 9B, everyone is saying 9B. Let's see. So this is the sporogenous tissue. Sporogenous tissue is being covered by tapetum because it provides nourishment. First layer. Then you have 2 outer layers. One is epidermis and then there is endothecium. In between epidermis and endothecium and the tapetum, you have a layer which is known as middle layer. So this is epidermis, this is endothecium, then middle layer, then tapetum, and then sporogenous tissue. So your answer is epidermis, endothecium, middle layer, and tapetum. B. B. So all of you got B correct. Okay, you are going really great. I will read out your names after showing you the next question. Okay. Which of the following is not a correct statement? Placenta is located outside the ovarian cavity. Please use your brain over here. Okay? Be very sensitive over here. Placenta is located outside the ovarian cavity. Central cell develops into PEC, that is primary endosperm cell, after fertilization. Orchid contain more than one ovule in an ovary. Egg apparatus consists of 1 egg cell and 2 synergids. You have to find what is not correct. Please, whenever you are solving correct, incorrect question, underline what you are solving. Some people just underline correct and come. No, please read not correct is also mentioned over here. Tell. So for 9B, who all said, let me see. Nirmala, Darshan, Ammu, Zahara, Samiksha, Riddhi, great. Shaukat Ali, Sparsha, Prateek, and Sinchana, Gulsita, okay. Then it is repetition. Neel, Aksar, okay. 10A, is it correct? It's not correct. Yes, exactly, guys. How did you spot it out? Good dose to you. So ovarian cavity, my dear children, is like this. This is the, oh my god. Okay, this is the ovary. Now over here you have the placenta. Placenta is the one that attaches to the ovule, providing it initial nourishment. So it is inside the ovarian cavity, not the outside. No, so it will be wrong. This is the not correct statement, incorrect statement. So 10A is the correct answer. Rest all of it is correct. Orchid has more than one ovule, okay, and egg apparatus, you know, central cell develops into primary endosperm cell. Great going. So again, read out the next thing and then I will be telling your name as well. Select the correct sequence of events in microsporogenesis. What is the correct sequence of events? I already solved this question. Then it's okay. Then you can go read out other things, okay? A, 11 answer, my dear children. For 10th, who all got the answer correct, kudos to you, my dear children. I'm seeing down your names over here. Let's see 11th. How are you, ma'am? That's so sweet of you. I'm good. Thank you for asking. I hope you also are doing great. So, correct sequence of events in microsporogenesis is basically you have sporogenous tissue. Each of the cells of the sporogenous tissue says that we can form microspores. So, they are all microspore mother cell, MMC. The microspore mother cell will undergo, this is 2N, it will undergo meiosis. Meiosis happens in 2 stages. First stage, it will have haploid number of chromosomes. This is meiosis 1. Then again it will divide, but this time no halving, it will remain like that only. So, haploid remains haploid itself. This is meiosis 2. At the end, you get 4 cells, you call them microspores tetrad. Tetrad as in 4, microspore tetrad. Each of them will be separating and give you microspores. Okay, each of them will be separating and give you microspores. So, your answer is sporogenous tissue, then microspore mother cell, then microspore tetrad, and then microspores, and then pollen grains as well. And then pollen grain, and then male gametes, and then male gametes. This is how it flows. So, A is your answer. So, who all got A? Kudos to you. Okay, going ahead with the next question that we have. Statement 1, very important question. Ma'am, last 3 chapters, what you have to do is solve questions. Solve questions, okay? 2 marker, 3 marker, 5 marker, and top 80 plus no. In that, the last questions are in part 3. It will be finished really, really fast. Okay, so don't worry. Ammu, that's so sweet of you. Thank you. Okay, children, tell me the answer for this. Apomixis is the production of seeds from unfertilized ovules. Is it correct? Production of seeds from unfertilized ovules? Yes, it is correct. Okay. Now, statement 2. Embryos produced from apomictic seeds are not generally identical to the parent plant. Wrong. Apomixis in embryo sac, no. You have these cells, okay, antipodal, synergids, everyone, basically they are haploid in nature. They have half the number of chromosomes. So, when the egg cell is there and all, when the male gamete comes, N and N fuses, you get 2N. But over here in apomictic seeds, somehow the meiosis has not happened like that, okay. So, they have 2N number of chromosome, the egg. So, does it need another N? No, it itself becomes what, my dear children? It itself becomes zygote. This is apomixis. And over here, since there is no male parent involvement, there is only one parent's involvement. So, the bacha that will be formed, my dear children, would be just, the makali that will be formed will be just like the parent plant. So, it will be identical. It will be identical, my dear children. So, your answer is A. Statement A is correct. Statement 2 is false. Embryos produced from apomictic seeds are not generally identical to the parent plant. This is a wrong statement. It is completely identical like clones. Okay. Going ahead to the next thing. The ploidy of perisperm present in beet seed is. Yes. The answer for this, who all gives me answer for this, I'll understand that basically you have read your NCERT really, really well. The ploidy of perisperm present in beet seed is what? Tell me the answer. I am drawing something. B, 13B, 13C, what are you saying? The ploidy, diploid, B, B, C, D, okay, A, B, C, D for me. Listen to me. All the cells of the embryo sac, my dear children, all the cells of the embryo sac except the central cell, basically they are haploid, having n number of chromosomes. Now, ploidy of perisperm, what is perisperm? I told you about nucellus. In the ovule, you have nucellus. You know what? One cell of the nucellus is the one thing that will go ahead, do megasporogenesis and later form the embryo sac. What happens to all the other cells? They usually get degenerated. But over here, the cells of the nucellus are going to provide nourishment. Okay, and this layer that is there for nourishment is known as perisperm. It's just a normal cell. This cell is just a normal cell, so it will have a 2n number of chromosome, my dear children, diploid number of chromosome. So your answer is diploid. Where do you find perisperm? Not all plants will have perisperm. Not all plants will have perisperm, my dear children. Only in beet. And one more. Can you give an example? Can you give an example for this? Perisperm is found in 2 plants. One is beet and the other one is, ma'am, L. Sudarshan is saying, ma'am, where can I get the PDF? Basically, you will get the PDF if you join the WhatsApp channel. In the WhatsApp channel, there is a link given for downloading the PDF. I will be uploading this PDF after I complete this class. Okay? 13B, 13B is the wrong answer. Yes, black pepper and beet is the thing. So beet and the black pepper, both of them start with B only. Both of them have perisperm still persisting. Usually we do not read perisperm because we have endosperm, we have cotyledons and everything. But over here it's there. So your answer is B, my dear children. Please, mango, Ammu, mango is an example of where, I'll tell you one thing. Mango and the citrus plants which are there, citrus fruits, that is orange, okay, they are example of polyembryony, where what happens, same things. What happens is that the nucleus is still persisting and the cells of the nucleus, the cells of the nucleus, they grow into the embryo sac, forming multiple embryos. Forming multiple embryos, that is known as polyembryony. So polyembryony is a very important thing. Example is mango and citrus plants. Okay. Oh my God, Sanjay, you're telling the scientific name also. Great. Zuhra, Sanjay, every one of you, Safura, great, great, great. Next one. The correct sequence of embryonic development in angiosperm is, yes. Can you tell me the answer for this? The correct sequence of embryonic development in angiosperm is, again, NCERT question. WhatsApp link is given in the description box. Sudarshan, in the description box it is given. So correct sequence of embryonic development is what? How does the embryo form? What do you call this process? Embryogenesis. The formation of embryo is known as embryogenesis. Okay. 14C. Okay, who are saying 14C? Let me see that. Nirmala Sutar says 14C. Okay. Rest of them have B and A as an option. Listen, first thing you have the zygote. First thing you have the zygote. Male and the female gamete fuses, you get zygote. Zygote will divide to form proembryo. Proembryo will form globular embryo. Globular embryo will form heart-shaped embryo. Heart-shaped embryo will be forming mature embryo. So your answer is C. So proembryo, then gametole, okay, and then it will be heart-shaped and then mature embryo. You remember much? Okay. Can you please tell the trick? I might have forgotten. So C is the answer for this, okay? Great going children who all are getting your answers correct. Awesome. And now we are getting ahead with the next chapter, that is human health and diseases. So we have completed almost 4 chapters which are high weightage chapters, right? 4 or 3? I think, yeah. Let's get ahead. Let's go. Okay. First one, which of the following pairs contains an infectious and a non-infectious disease properly? Easy question. One is infectious, that means it can be passed from one organism to the other, okay, and the other one is not infectious. Which one of is that? Leela, 14C, yeah baba. I'm just not reading out because I don't want to waste the time. But yes, I want to appreciate you, which is why I read in between, okay? I'm so sorry. Which of the following is easy, tell me. Which of the following contains this thing? 1, A, B, C, D. Sparsha says B, Ammu says B. Yes, the answer is B. Answer is B. A, children who are saying A, can you just check once? How can it be A? Typhoid, it spreads. AIDS also is an infectious disease. It's a sexually transmitted disease. It's infectious. You can get it from other organism. But cancer, can we get from other people? No. So AIDS and cancer is the thing. AIDS is infectious and cancer is not. Got your answer, my dear children. If you have made it wrong, please. Now, an intestinal parasite which causes blockage of the intestinal passage and whose eggs are excreted along with the feces of the infected person is that what? So there's a parasite that affects, just a second, okay, that goes and affects the intestine, my dear children. What is that particular parasite? And moreover that, it lays the egg and the egg is released to the fecus of the infected person, as in 2 number. It is released to the 2 number, okay. So what is this? The answer for, tell me like this, 2A, B, C, D, otherwise I'm not gonna take it. A, B, A. Children, please write 2 in front. So every one of you who are saying B, you are correct. It is Ascaris. Okay, Ascaris is a nematode. It's a Aschelminthes and it is the one that causes the whole trouble. Ascariasis. Okay, so B is the answer for this thing. Vocularia bancrofti, it causes filariasis that affects your lymphatic vessels. Epidermophyton, it causes ringworm disease. Microsporum also causes ringworm disease. Next thing. Ma'am, lag is there. Okay. Children, now it's fine. Okay. Calm down, calm down. I think it's fine now. Can you just reload it once for those children who are watching this video? I think there was a glitch over here also, I saw it. So can you just reload from the starting? I mean close and open. Now no lag. Okay, continue. Okay, chalo. So the answer is Ascaris. Okay, going ahead with the next thing. Which of the following is affected by the infection of Eukaryotic bankrafti? Which one of them is affected by Eukaryotic bankrafti? I just told you. Eukaryotic bankrafti is causing filariasis or elephantiasis. And why do you get elephantiasis? Because what happens is that the fluid, the extra fluid that is there in your blood vessels, it oozes out and basically it goes around the cells. It just stays around the cells, leading to inflammation. Leading to what? Inflammation, my dear children. Okay. So where is it actually affecting? It is the lymphatic vessels. Because why is this fluid getting together over there? Because the lymphatic vessels are damaged. Lymphatic vessels are the one that drains out this fluid. So it does not allow inflammation to happen. If lymphatic vessels itself are damaged, then who will drain out? Hence, this disease is basically lymphatic vessels. Ninanda, hi. Great going, everyone. No fact, no fact. Told the correct answer. And all the other children, Ammu, Deepak. Children, please read the fourth question as well. Which of the following pairs correctly matches a disease and a pathogen causing it? Now let me read out the names. Okay, so we have Sanjay, we have Vriddhi, I don't know computer, certain videos have been put up, please watch those. Okay, Ashok, and then Ammu, no fact, Sanjay, Janika, Shaukat Ali, Samiksha, thank you for saying, and then Chinmay, Ninanda, Safura, all of you, thank you my dear children. Okay. I don't know, for computer video you cannot find it here, okay, please. So I'm so sorry, but yeah. So fourth answer, you have to find which is the correct match. So no fact says A, Riddhi says B, and Darshan says B, so let's see. It is A, which is correctly matched. Ringworm disease is a fungal disease. Why it is known as ringworm? Because they have, the people who have this disease have a lesion like this, like they have an outgrowth like this. Okay, so when you touch it, you can feel it. It looks like a worm, but it is not a worm disease. It's a fungal disease, and it is caused by trichophyton. So there are three things: trichophyton, epidermophyton, and microsporum. Three of them are the one that causes ringworm disease. It is correct match. Talking about the people who said B and C, children, pneumonia is caused by Haemophilus influenzae, not pneumonia. That is Streptococcus pneumoniae. It's wrong. These two words are wrong. Malaria is caused by Plasmodium, not Ascaris lumbricoides. Ascaris lumbricoides causes ascariasis. Ascariasis is not caused by Salmonella typhi. Salmonella typhi causes typhoid. Okay. And malaria is caused by, yes, Plasmodium vivax, falciparum, P. malaria, great genus. And Janika, sorry. And now we have what over here? For children who said the A answer, all of you, good going. Okay. Next question, fifth question. Hemozoan is what? OMG, Safura, why there is an OMG over here? Hemozoan is by what? Hemozoan is a precursor of hemoglobin, toxin released from Streptococcus infected plants, toxin released from Plasmodium infected cells, not plants, okay. Toxin released from Haemophilus infected cells, my dear children. What is the answer for this? Fifth answer. Fifth C, yes, it is the toxin released from Plasmodium infected cells. So Plasmodium, it goes and affects our RBC. In our RBC, there is hemoglobin, and it breaks down hemoglobin to heme and globin. But the Plasmodium does not like this heme, so it basically converts it into hemozoan. It converts into hemozoan, and this is the toxic thing. So when RBC bursts out, it becomes toxic for us because it is being released. So your answer is C, not D. It is Plasmodium infected cells. Okay, Plasmodium. So malaria, in malaria it all happens. So 5 is C, not D. Children, please get it correct. And no need to delete your answers, it's fine. Okay. Next question, hi Darshan. Okay, next question, the human immunodeficiency virus primarily attacks and destroys what? HIV virus. What does it primarily go and attack and destroy? What is that particular thing? Why not D? Because it is not Haemophilus. Haemophilus is basically happens in Haemophilus influenzae is the one that causes pneumonia, and in pneumonia you don't have hemozoin. Malaria is the one where hemozoin is being released. That is why we get fever and chills. Okay. Yes, tell me the answer for this. Ma'am, RBC release that toxin. So as I said, RBC is there. In the RBC, the hemoglobin is broken down to heme and globin by the plasmodium itself. Okay. Now heme is toxic for the organism, so it converts into hemozoin, which is actually toxic for us. So when the RBC breaks open, the hemozoin is released, which is why we get fever and the chills. Okay. Okay. The human immunodeficiency deficiency virus primarily attacks and destroys. The answer for this is B. The answer for this is B, my dear children. Okay. Th lymphocyte, that is T helper lymphocytes. T helper lymphocytes are the one that the HIV virus comes and attacks. One very important question, my dear children, one very important question is that bispersha, please definitely go and pray for us, everyone. Okay. Now, okay, I was saying about HIV virus. So HIV virus basically it divides itself, it increases the number in the macrophages. That is why it is known as HIV factory. It increases the number there, but then it goes and affects the T helper lymphocytes. Very, very important thing. Okay. Hi Kavita. Okay. Going ahead with the next question. That is this. Which of the following is an autoimmune disease? A, rheumatoid arthritis, B, tuberculosis, C, malaria, D, AIDS. Bye for now, ma'am. I'll watch. Yes, definitely. No problem. Can you check 5 Hawa? Which of the following is an autoimmune disease, my dear children? You're asking about 5th question. I explained 5th question, please. It will come to you slowly. 7A, 7A, 7A. Yes, it is 7A. Autoimmune disease, what do you mean by that? That means our body is not able to identify which is our cell, which is not. And it goes on killing our own cells. That is what is known as autoimmune disease. An example is rheumatoid arthritis. Yes, Captain. Hi, Praju. Okay. Okay, my God. Now, question number 8. Antibodies pass through placenta and is present in colostrum, which protects the newborn from certain diseases is. You have to tell the antibody which is passed from, from the placenta and also from colostrum. Dimple, please go and have food, Baba. No problem. Being healthy is more important. Go have food and then come back. Okay. Hi, Sahana. Children, placenta from the mother. So someone says B, Dhanush says B, Leela says B. Okay, from the placenta to the baby, it is IgG that is transformed. Okay, and now from the colostrum, that is the first breast milk, IgA is transformed. So I always remember like this, G-A-M-E-D, gamed. Okay, this is the order. G is the one that passes first, A is the second one that passes, and then M and D, let's not talk about it, it's not there in your NCERT. That is the function, and E is released during allergy. E is released during allergy, remember like that. So your answer for this question is B. So via placenta, via milk. So your answer is B, not C. Okay, check your answers. The site where lymphocytes interact with antigens and proliferate to become effector cells are. What are these sites known as? Where the lymphocytes, the T lymphocytes and B lymphocytes, they see the antigen and now they proliferate, they increase the number and they do dishum. They become effector cells. What is this site? Can it be, lymphoid organs, there are 2 sites, primary lymphoid organs and secondary lymphoid organs. So in primary lymphoid organs such as thymus and bone marrow, what happens is that they are just formed. B and T cells are just formed and they mature. Bone marrow is a place where everything is being formed. B lymphocytes stay here only for maturation, T lymphocytes go there for maturation. So they become antigen sensitive. We say that they become antigen sensitive. They know that they will be able to recognize that antigens are there. Now they don't do the work over here. They go to secondary lymphoid organs such as spleen, such as lymph nodes, such as Peyer's patch, such as tonsils, and do their action there to become the effector cells. So you have to look for secondary lymphoid organs, not the primary ones. So thymus and lymph nodes, wrong, because thymus is a primary one. Bone marrow and thymus, both of them are primary ones. Peyer's patch and tonsils, this is entirely correct. Bone marrow and spleen, again wrong, because bone marrow is there. So your answer becomes C, my dear children, not B. Not B, C, C, C. Understand the question. Here, they see the antigen, they increase the number, and they become effector. They do dishum. So it happens in secondary lymphoid organs, not in the primary ones. In the primary ones, they're just maturing, they're just forming, and they're just maturing to become antigen sensitive. If the term comes antigen sensitive, just understand you're talking about primary lymphoid organs. If the term comes effector cells, that means you're talking about secondary lymphoid organs. Okay? Cool? Shavani saying 12 after 15 will jayenge. Arey, you'll get full on full. Don't worry. Question number 10. No need to be sorry. It's okay. So, hepatitis B vaccine is produced from. Again, this is a question very important. I made it. This is an NCERT based question. Okay. Hepatitis B vaccine is produced from. In your human health and diseases, just below, the people who are there with me throughout, okay, who have heard my lectures before also, they know the answer for this. So hepatitis B vaccine is made using a transgenic organism. That means we are going to change some organism, and with the help of it, we are going to make a vaccine. So the answer for this is yeast, my dear children. I'm not wasting too much of your time. Let's not do the guesswork. Okay, it is yeast through which you get the hepatitis B vaccine. If you do not know the answer, learn it right now. Okay, it's given beneath the vaccination and immunization paragraph. So 10B, yes Sinchana, yes Dhanush, no fact, Leela, everyone, correct. So don't get this answer wrong if this question comes. Now question number 11, which drug is used as a medicine to keep patients, to help patients with depression and insomnia? As in, if they are not able to sleep and they are depressed, what is the medicine or drug that is given? Any guesses? Yes, Praju, tell me. You can ask the doubt. Children, 11th, A, B, C, D, what is the answer? Hi, Rocky. Timings are. A, Dhanush says A, Riddhi says A, Leela says B, Ayu says A, children. It will be B. It will be B. Morphine is used as a painkiller and sedative. You want to make people sleep, you can give morphine. Don't give, doctor will give. Okay? Painkiller as in they have surgery and all, pains a lot, right? So that for that time, for them to heal, you need them to sleep. That time morphine is given. But for fighting with depression and insomnia, you have two drugs, barbiturates and amphetamines. This becomes your answer. B is the answer. Raghu, please reply. We are CS students. CS children, my dear children, I cannot teach you CS. Otherwise, I would have definitely done that. Okay, so the ma'am who was available made a few videos and it's there in the channel. Okay, that's the only thing we could do for you guys. Okay, children, I hope you got the answer. Going ahead with the next question. A chemical structure is given below. Identify its plant source. Oh my God, Naveen is so fast saying C is the answer. Is it? 12. What is the answer? If you're not able to see the image, I'll just zoom it in and show it to you. A chemical structure. This is the chemical structure, my dear children. You have to find and tell what does this belong to, which plant gives it. Darshan. 12C, 12C, 12C. Yes, it is Papaver somniferum. Papaver somniferum gives you two drugs. One is morphine and the other one is the acetylation version of morphine. That is what? That is heroin. So this is the structure of morphine. How do I remember it? Both of them are known as opioids. Why? Because Papaver somniferum is also known as opium poppy. So that's why opioids. So this looks like an O. So this is morphine, which belongs to opioid family. Okay, so this is the answer. Got it? Next question. Let me read out the other answers. Until then, you please read out the question. Okay, so Darshan says C, Dhanush says C, Hamza says C, great, Chinmay C, Ammu C, Harinakshi C, Diagram is not visible. I hope you saw it now. Not fact. C, Riddhi and Aditya. C, Aniketan. Oh my God. Sinchana. Fortune Speed. Ninanda. Aditya. Amrita. Oh my God. A lot of you said C. Great. Dhanush now says B. Charas and ganja are the drugs which affects what? Respiratory system, cardiovascular system, digestive system or nervous system. First things first, tell me, charas and ganja belongs to what? Charas and ganja belongs to cannabinoids. Cannabinoids. And cannabinoids are derived from cannabis sativa, okay? And they are going to affect the cardiovascular system. They are going to affect the cardiovascular system, your heart and everything, your blood vessels and everything. Please make sure you're not making mistake. If the question comes, the drug that affects the blood pressure is not cannabinoid, it is nicotine. It is nicotine which you get when you smoke. Okay, so that increases the blood pressure because nicotine makes the adrenal glands release adrenaline. So this is cardiovascular system, that is cannabinoids, both of them. Okay, just to avoid confusion, I told you the other thing as well. Okay, works? Children, can I take 1 minute of your time and explain something very, very important? That's why. Okay, you have opioids, you have cannabinoids, and you have coca alkaloids, that is cocaine. Okay, opioids basically affects your CNS, central nervous system, and also your gastrointestinal tract, GI tract. Okay, cannabinoids affects your cardiovascular system, coca alkaloids, that is cocaine and all, affects again your CNS. So both of them affecting your CNS. This one gastrointestinal tract as well, and this is cardiovascular system. In CNS, it is going to affect the dopamine transport, which is why if someone has cocaine, they will be having a sense of euphoria, too much happiness, okay, because dopamine is a happy hormone, things like that. Okay, can I go ahead? I just sort of telling it to you because there are high chances that you might forget. CNS stands for central nervous system. And one more things, for hallucination you have coca alkaloids. You get hallucination, you'll see cow is flying, you are flying and everything. Hallucination happens in coca alkaloids. Apart from that, hallucination happens in two more things, atropa belladonna and datura. Atropa belladonna and datura. Okay, good, got it? Yes, Sanjay, you get it from erythroxylum coca plant. Exactly. Children, please talk to me, do not talk to each other. Don't waste your time. After that, we'll enjoy later on. Okay. 14. Smack is a drug obtained from latex of Papaver somniferum, leaves of cannabis sativa, flowers of datura, fruits of erythroxylum coca. Smack. You should know the other name of the drugs as well. For example, cocaine is known as coke or crack. Coke or crack. So you get it from erythroxylum coca. Okay, now you're asked about smack. What is smack? From these questions, anything, concept, anything can be asked, my dear child. Okay, so please watch the videos that I put up for you. It can be long. Put it in 2x speed and watch it. Very, very important. So Naveen says A, Shahma says A, Sumaya says A. A lot of you says A. It's in heroin, there is no E, okay? Okay, smack is a drug that is heroin, my dear children. Exactly, you all are correct, and you get it from latex of Papaver somniferum. The parts in that you get milky white latex, from that you get this. So this is your correct answer, and all of you who said this, great going. Okay, awesome children, awesome. Very proud of you. Next thing. Interferons are most effective in making non-infected cells resistant against the spread of which of the following diseases in humans? As in, the question says, if this cell is releasing interferons, what does it tell to the other cell? That you have to be same from what? That you have to be same from what? That is the question. Who is heroin? No one is heroin. Okay, heroin is a drug. Heroin is a drug. Oh my God, children. Tell me. 15th. I believe all of you are good bachas, right? All of you are good children. So please don't talk to yourself, Baba. Don't waste anyone's time. 15A. 15A. Is it correct? 15A. Why did you say 15A? Because, my dear children, it is the virus infected cells that releases interferons and tells the other cells that, hey, virus attack is there, you have to be safe. So which of them is a viral disease? It is AIDS. It is what? AIDS. AIDS is a viral disease. So this becomes your answer. Okay, great going. For other children, amoebiasis is caused by a protozoan, ringworm is caused by a fungus, ascariasis is caused by a nematode or Ascalmenthes. So your answer is A. The human host cell in which the gametocytes of malaria parasite develop are, very important, very important question, very important concept as well. The human host cell in which the gametocytes means, you know what, plasmodium, the vector which is there, it has multiple stages. It has different stages. One stage is gametocyte stage, where it will now divide and form the gametes. So it has not divided yet, it has just a gametocyte, it is in the gametocyte stage. So in which cell of the human host, human body, is this particular parasite going to form the gametocyte stage? That is the question. So 16C, everyone is like first, okay. The answer is C, yes, erythrocyte, that is RBC. In RBC, the gametocytes are going to be formed. But do they form gametes in the human body? Very important question. Do they form gametes in the human body? Where does the fertilization of gametes happen, of plasmodium? Where does the fertilization of gametes happen? Tell me this also. If you know the concept, tell me this also. The gametocytes are formed over here. The gametes are not going to be formed in human body. In which organism's body is the gametes going to be formed? Gametocytes, my dear children, are going to be released in the blood. Who drinks our blood? Who drinks our blood? You will say Jasmine ma'am. It's not Jasmine ma'am, it's what? It's female Anopheles mosquito. And now, when the gametocytes reach the gut of the female Anopheles mosquito, there it will divide into male and the female gametes, they will be fusing together. So fertilization happens in the gut of the mosquito. Okay? Ma'am, teach how to draw diagram. I don't know if it's possible with this short duration, but I'll try to, okay? I have put the most important diagrams though. Yes. Next, match the man with the, oh my god, why is the evolution question there? Okay, just a second. Chalo, let's solve one evolution question also. It just came randomly, but you should be knowing. You are someone who is preparing for biology, right? So all of you said mosquito got great. Anopheles mosquito. Children, the mosquito is always female, okay? The one that drinks our blood is the female one. Now match the type of man with their origin periods and choose the correct answer. It's evolution chapter, okay? Don't get scared. It's not in human health, but still. B. Everyone says B. Is it B? So listen to me. I'll tell you a story very fast. You will be able to remember it. First, we have two organisms, Dryopithecus and Ramapithecus. Dryopithecus ape-like, okay? Ramapithecus man-like. Ramapithecus and Dryopithecus came 15 million years ago. Ramapithecus was more man-like, name is Ram, right? And then from them came Australopithecus. From them came Australopithecus. When, my dear children? Almost 2 million years ago. Almost 2 million years ago. They had stone weapons. They had stone weapons for killing down things. But what did they kill down? Did they eat non-veg things? No. They were vegetarians. They were seed-eating, fruit-eating organisms, my dear children. Now from Australopithecus came actually Homo habilis. It is not mentioned over here. Came Homo habilis. Now from Homo habilis came Homo erectus. They stood erect like this, okay? And which year? Almost 1.5 million. This was 2 million years ago. This was 1.5 million years ago. Now, from Homo erectus, some organism went to different location, and there they formed Neanderthal man, my dear children. They started feeling shy. They covered their body, and they used to bury their dead also. So this was comparatively recent. So it's like 1 lakh to 40 years back, okay? Homo erectus, I told you, some of them formed the Neanderthal man, some of them gave rise to the modern Homo sapiens that we are today. You get it? That is around 75,000 years back. So this becomes the answer. So A goes with 2. A goes with 2. So this is the only option that is there. So B answer is correct. So B is there? Yes, Chinmay. Good boy. Okay. So dear children, your answer is B. I hope you got this correct. Please do not get it wrong. Now, a farmer working in a field was bitten by poisonous snake. Doctor, I think this is the last question of human health and diseases, okay? Now, Farmer working in a field was bitten by poisonous snake. Doctor gave him an anti-venom treatment that contained preformed antibodies. This type of immunization is known as. My dear children, whenever you get ready-made antibodies, that means your body is not participating in anything. It's not doing anything. You get it? It is passive immunity. It is passive immunity. When your body gives antibodies, that's when you call it active immunity. So this is passive immunization. This becomes what? Passive immunization. Yes, you are correct. C is the correct answer. Chinmay, Dhanush, Ammu, no fact, Shahma, Ashok, so great. Riddhi, everyone of you, awesome. Samiksha, I hate myself. Okay, so this is passive immunization. Again, a question for tetanus toxoid. If tetanus toxoid is given, again you are giving preformed antibodies. This is also an example of passive immunization. In vaccination, my dear children, opposite of immunization, what do? You give weakened or disarmed pathogen to the body. The body will produce antibodies. Next time when this guy actually comes in, your body will fight back against because it has the memory. So that is active immunity. Vaccination, you have active immunity. Prajwal, ma'am, you're still energetic after being, thank you so much. Do I have an option? No, you guys will get bored if my energy goes down. Okay, now we have human reproduction. Are we ready for this chapter as well? Are we ready? My dear children, don't think that we are losing on time. It's very important for practicing MCQ. If you're someone who has prepared a lot of things, you might be understanding a little of concept also here. Okay, now if you're someone who have not touched your NCERT, go read your NCERT. I don't want to be you to be there in the live. Okay. These questions are from the question bank and also the model question paper that the government has released. My dear children, 51 of them who are here over here, please make sure you are going and reading the model question paper answers and then only going for exam. Okay, question number 1, scrotum. Ma'am, if possible, can you explain about innate immunity? Innate immunity means you have it from your childhood. When you are born with that. For example, skin. For example, tears, everything. You're born with that and it's non-specific. For example, a virus tries to get inside, you have the skin only. Bacteria also, you just have the skin. The skin is not producing anything extra for the bacteria and virus. So it is non-specific type of immunity. So you have different things. You have physical barriers, okay, such as skin, physiological barriers such as your tears, and then cellular barriers such as certain cells which are there. These cells will be eating up anything that is foreign. It is not specific, such as PMNL, polymorphonuclear leukocytes. Okay, so all this thing and also macrophages, all this thing. And then you have interferons, that is the last one. Cytokines, that is the last barrier. Okay, now let's get ahead. Okay, my dear children, scrotum helps in maintaining the temperature of testis, which is lower than the normal body temperature. How many degree? 2 to 2.5 degrees Celsius lower than the normal body temperature. Okay, so this and this can be the answer. I'll cross it out. Now, each of the testes has about B compartments. What compartments? Testicular lobules. How many? 250 of them. So this and this again correct. Now measure C centimeter in length. What is the length of a testis? 4 to 5 centimeters. So this becomes the wrong thing. This becomes the right thing. B is your answer. Okay. B is our answer. Come on, Captain, drink some water. I'll drink. Thank you. Children, I hope you're also drinking some water. B is the answer. Let's go ahead with the next question. The function of myometrium layer present in the uterine wall is to. What is the function of myometrium layer? You have 3 layers, perimetrium, myometrium and endometrium. Okay. What is the function of the middle layer, that is myometrium? That is a question. And my dear children, if you're liking this video, okay, definitely like and also subscribe and suggest to your friends, ask them to join, and also comment down if there is something really bad or good that you want to talk about me, or if you have any doubt, still you can post it down. Okay. Yes, idralle MCQ barta ida. But you have to go through your NCERT as well. Okay. I have not studied anything. Okay, I'll come back to you. Okay, Guru Kiran, I'll come back to you. So the function of the myometrium is to basically undergo strong uterine contraction, because if suppose baby is there in the uterus, the baby has to move out. For that, basically there should be contraction, and myometrium is a muscular layer made up of smooth muscles. Smooth muscles are not in our control. I cannot think that smooth muscles can contract and relax according to my wish. It is an involuntary thing. Our brain controls it. So it exhibits strong uterine contraction during parturition, during expulsion of the baby. So this is the thing. Undergo cyclic changes during menstrual cycle. It is not the myometrium, it is endometrium, the innermost layer. Gives protection to the uterus, it is perimetrium. Helps in implantation process, again endometrium. Okay. Now going ahead. Seminal plasma in human males is rich in what? Very important question. Tell me. Ma'am, please explain about perimetrium. Perimetrium is the outermost layer which is there. It just provides structural support and also protection to the uterus. Okay. Ma'am, these MCQs are coming in the exam. I hope so. I hope so. I definitely hope so. Obviously, direct questions might not be coming, but in and around they might come. Okay. And Guru Kiran, if you have someone who have not studied, I'll recommend you to please watch 2, 3 and 5 marker videos, the previous year videos, and then previous year question videos, and then please read the read the question bank answers. You can find the question bank answers in the WhatsApp channel. Go to the WhatsApp channel. There's a link that has been put up for the PDF. Go read it. I have made 3 parts, top 80 plus questions. All the questions are taken from the question bank. Okay. Seminal plasma is in human base is rich in fructose and calcium. B. It is, the answer is A. So all of you who got A answer, you're absolutely correct. Why? Seminal plasma is made up of secretion of 3 glands. First one is seminal vesicle, second one is prostrate gland, okay, prostrate gland, and the third one is bulbourethral or corpus gland. Okay, now seminal vesicle releases fructose. Why? Because it wants to nourish the sperm, it wants to give energy to the sperm. Prostate gives calcium and clotting enzymes. Certain enzymes are also released from prostate. Bulbourethral glands helps in lubrication. All the secretion together, you call this as seminal plasma. So over here it is fructose and calcium which matches everything. There is no glucose. Please remember this: fructose, calcium, clotting enzymes and lubrication. That is the function. Okay, seminal plasma plus sperm is equals to semen. So your answer for this question becomes A. Cool? Yes. Okay. Great, Shagufta, awesome. And everyone else, Kalkapa, Ammu, Sumaya, I'm so sorry I cannot read your things. I'll drink some water. By then, children, please solve the next question. Ninanda, Sumaya, Riddhi, Lingaraj, Sat, Sahama, Shagufta, Varsha, Samiksha, Vidya, Chinmay, Ramesh, Dhanush, Riddhi, Prajwal, everything. Fructose formula would not be asked. This is not chemistry paper. Da. Okay. May Vishwas. Okay, so Vishwas. Tell me the answer for this. Children, please drink some water as well. Fast, ma'am. I'll die if I go more fast than this. Okay. 4th question. B you are saying, D you are saying. Okay. The process of release of sperms from the seminiferous tubules is called spermatogenesis. Is it called spermatogenesis? For those of you who are saying it is called spermatogenesis, you are wrong. Okay, the process of release of sperms from the seminiferous tubules is known as spermiation. Very important question, conceptual question also. They can ask you to explain spermiation. Okay, now the spermatids are transformed into spermatozoa by the process called spermiogenesis. Spermatids are transformed into spermatozoa by a process called spermiogenesis. Is it true? Yes, it is true. So I'll say statement 1 is false, statement 2 is correct. Statement 1 is correct, incorrect. This is the answer for this. You're so fast. Now Ramesh is saying I'm so fast. Children, listen. The process of release of sperm from the seminiferous tubules is not known as spermatogenesis, it is known as spermiation. Always remember, aviation, it is released like that. Okay, you can remember, spermiation. Now, listen, when you form sperms, initially you have spermatogonia. Some cell, they do undergo mitosis and they differentiate to form primary spermatocyte. They form primary spermatocyte, which is 2N thing. Children, I'm just explaining because you might find similar questions now on. Okay. Now, they divide, they undergo meiosis number 1. Now you get, from primary spermatocyte, you get secondary spermatocyte. And the number of chromosomes will be haploid. Again, the secondary spermatocyte will divide into 2. This time it will be having N only. There is no more halving. Okay, that is N. Now over here, my dear children, what do you call them? They are like kids. They are immature. So you call them spermatids. You call them spermatids. They are like kids. Now you want to mature them? They are like globular. But is the sperm globular? No, it is long, right? So to mature them, you need to convert them into spermatozoa. This process is known as spermiogenesis. and that is exactly given over here. So this statement is correct. Now the spermatozoa is made in the seminiferous tubules. When it is released, it is known as spermiation. Got it? But this is the wrong statement. It is written as spermatogenesis. The whole process is known as spermatogenesis. Okay, so I'll say D is the correct. I hope you got it. Thank you so much, Shambhavi. Ma'am, please explain first question. First, just a second. First one. Are you sure? Tell me the chapter. I'll explain it later, okay? We are gone ahead. Okay. Fifth question, very important, very important. Ali, thank you so much, please. Thank you so much. For 3 markers, which all... I'll come to you, Sat, okay? Please try to read this question, my dear children. To produce 2000 sperm and 400 ova, how many spermatogonia and oogonia are required? How many spermatogonia and oogonia are required? Do you know? Shagufta says B. Oh my God. Ammu says B. 5D. Okay. Children, thank you. Thank you, Blades and Ali and everyone. But let's be focused on our studies. Yes? You all are so beautiful. Okay. Now, ma'am. B. Okay. So, lots of you people are saying B. Let me keep B as an option. So, listen. For 2000 sperms from one primary spermatozoid or basically spermatogonia, you are basically getting 4 sperms at the end. So 2000, if they are there, sperms are there, then 2000 divided by 4 is equals to what? 500 spermatogonia. Now talking about oogonia, you know about oogonia, there is one primary oocyte, it divides into 2, but there is one cell that is polar body and this one is the secondary oocyte. Okay, so this does not undergo any division because it might get degenerated. This polar body gets degenerated. Now meiosis 2 happens. Again, there will be second polar body and there will be one ovum. So from one oogonia or basically from one primary spermatocyte, you are just getting one ovum. So over here 400 ova are there. That means how many oogonia will be there? 400 itself. Your answer is D. Okay, got it? Cool edits. Yes. Okay, children, understanding? Now, I am pretty sure you will be able to answer this question. Question number 6. The release of sperms from the seminiferous tubules is called what? The release of sperms from the seminiferous tubules is called what? B. Ashok says 5B, Prajwal says B, Ali says B, Y vision A. I just explained. Release of sperm, aviation, spermiation. It will be spermiation, my dear children. It's A. Ma'am, for hormones secreted by placenta, should we involve? No, you should not involve relaxin. Relaxin is produced by ovary, not Kalkappa. Thanks. Thank you so much. Okay, going ahead, children. We have to finish, right? We have to finish. Yes. What is the correct sequence of sperm formation? Can you tell me what is the correct sequence of sperm formation? We just studied it again. So Shagufta, let me read out the answers, okay? Who all got the answers correct? Vibhishan, Sath, Ammu, Shagufta, Kalkappa, Samiksha, Shashikala, Sahama, Prajwal, Ali, Prajwal, not fact to fact, Vishal, sorry, yes. Okay, 7B. Vishal, okay, everyone says B, except I saw 1A, okay, except 1A. So over here, my dear children, what is the correct sequence of sperm formation? First you have spermatogonia, then from them you get spermatocytes, primary then first and then secondary, then spermatids and then spermatozoa. So your answer becomes B. Great going, awesome. I have got the best students over here. Children, now, which among the following has 23 chromosomes? Again, use your brain. Which among them has 23 chromosomes? That means half the number of chromosomes. 23 chromosomes, spermatogonia, zygote, secondary oocyte or oogonia. Please can you tell me the answer like this, 8 A, B, C, D? It becomes easy for me to understand. I'll say yes, your answer B is correct, but it might be, you might be talking about 7th answer. Ma'am Vishwas, yes. I have Vishwas on you, Vishwas, and I remember your name. Okay. Children, 8C, yes. Y Vision is a good person, said 8C. 8C, Y Vision, cool, it's C. Which among the following has 23 chromosomes? Yes, it is secondary oocyte. Spermatogonia has 2n number of chromosomes. It has not even started meiosis, right? Zygote is formed from fusion of n and n, 2n again. Oogonia, basically again, it has not started division, so it will be having 2n. Only secondary oocyte is the one that has n, half the number of chromosome. So your answer is C. Great going. Yes, definitely Shishir, I will. So question number 8 is C. Okay. Yes, Captain. Go have lunch and come. Match the items given in column 1 with those in column 2 and select the correct options given below. Very important question, and this will make you understand the concepts of menstrual cycle as well. So you have 3 phases in menstrual cycle, or basically 4. Okay, you can take that. Okay. Ninth answer. Hi Shripad. In proliferative phase, what happens? Something is going to proliferate. What is going to proliferate, my dear children? It is the endometrium that is going to proliferate. And while the endometrium proliferates, thinking that yes, fertilization can happen, someone else is also being formed, follicles. The primary follicles are going to, the secondary follicles are going to mature into what? Graphian follicle, to release the secondary oocyte within them. So proliferative phase and follicular phase works hand in hand, they together move. Okay, now what happens after this? Ovulation happens. After this, ovulation would be happening. When ovulation happens, corpus luteum is being made from the Graphian follicle. You get corpus luteum, my dear children, and this corpus luteum will be responsible for the formation of more endometrium. And this endometrium, thinking that fertilization will happen, I should be secreting some important things. It is known as secretory phase. And secretory phase is also known as luteal phase, because as I said, corpus luteum is being formed. Okay. Now menstruation is basically when the blood flows down. When the blood flows down, that means the endometrium has been broken. Okay, it is shutting down. So since you are saying C, let me see C. C says A goes with 2, okay. B goes with 3, okay, and C goes exactly. Awesome. I have mentioned B. Oh, that's wrong. Don't worry. Your answer is this only. Typo error. Hi Kavita. Okay, shall we go to the next question? Question number 10. Children, I will be reading out who all got it correct, but you can solve. Which of the following hormones is produced only during pregnancy by the ovary? So someone asked me right now, right? By the ovary. Prajwal. Yes. NEET classes, I'll check through, but KCET will be there definitely. Okay. So, hi Sharad, okay, Y Vision says it's D, Kalkappa says it's D, Vishal, Kavita, Cool Edits, Y Vision, Ammu, Noor Fatima says it's D, everyone says it's D, the answer is D, yes. Okay, so basically the muscles around there, the uterus, it has to be relaxed so the baby can be moved out. So ovary is the one that releases relaxin. Ovary releases one thing during later phase of pregnancy, and that is relaxin, okay. Is KCET free in YouTube? Yes, a lot of master stroke and everything will be free. But again, if you want a approach like Paka approach, obviously there is Abhyas KCET test series where the teachers who are there preparing children for KCET for years have prepared the question papers. It will be a lot more helpful. Okay, now if you are not able to pay, it will be there in YouTube as well. Okay, 10th D is done. Sorry. Okay, choose the correct option from the table given below for the formation and dissolution of the labeled part in the given diagram. So this labeled part, my dear children, you know what? It's not actually, okay, just a second, if you're not able to see this, I'll just zoom this in. Now it's visible? Yes. So labeled part is this, that is zona pellucida. You know what? This is not ovum, actually it is secondary, we can think of it as secondary oocyte. Okay, secondary oocyte is still not divided, suppose. So secondary oocyte is the one that releases zona, that forms the, it's not zones pellucida, it is zona pellucida, okay. It is being formed from that. And then later, so secondary oocyte is the one that forms it, but later what happens, this zona pellucida will be degenerated, it will be dissoluted. Okay, it will be dissoluted after fertilization, not before fertilization, children, after fertilization. Slowly the sperm will coming in, okay, they will bind to the receptors of the zona pellucida, then slowly they will be getting in. When they fuse with the secondary oocyte, basically fertilization completes. At that point of time, ovum is being made, ovum and the sperm fuses, and slowly the zona pellucida will be dissoluted later on. Okay. Ma'am, Janika, yes, Janika is back. Tell me, what is the doubt? So your answer is secondary oocyte and after fertilization, not before fertilization. Children, you are able to understand this? T, ma'am, please tell the live session. Prajwal, hi. Is it enough if I watch 80 plus question series? 80 plus question series will be solving all the questions from the question bank. Right, so it will be very much helpful for you, but still I'll recommend you to read through at least read. If you do not have time, just read through the 2 marker and 3 marker, because it contains the PYQs, right? PYQs and this. So just read it once. Okay, so all of you said D is the correct answer. If you said B is wrong. Okay, it is not formed in primary oocyte, it is from secondary oocyte, and it is formed dissolved after fertilization. Now, going ahead, one very important thing. Listen. When fertilization happens, for example, there is a sperm, the sperm comes and attached to the ovum, sperm come and attached to the zona pellucida. At this point of time, it allows the entry of one sperm. When the one sperm gets inside and tries to fuse with the secondary oocyte, my dear children, at this point of time, slowly certain chemicals are being released which makes changes in the zona pellucida layer such that it does not allow the entry of certain sperms. This is not what you mean by dissolution. Dissolution means it will totally go away, and that happens after fertilization. Okay. Going ahead with the next thing. During pregnancy, the fetus develops limbs and digits. Very important thing. You should know what is the time for each things. Okay. Michelson and Stahl experiment. Yes. Children, do you want me to stop this particular session, MCQ session, after this particular chapter? Or do you want me to complete all the high priority chapters? My God, no one is answering what I am telling. Question, children, all of you, please do not put your statements like that. Okay, it will be really bad. I am reading it down. Please, cool edits, Mohammed Amir. Wait. Okay. I have a straight question. Do you want me to complete all the other 3 chapters also, microbes in human welfare, evolution and biotechnology principle and processes? Those all are 7 marker and this chapters. Okay, do you want me to do that, or do you want me to stop and take up your doubts? Want every lesson MCQ? Okay, we'll do that then. Okay, what is free? Is it free? YouTube KCET series is free, my dear children, and number of recombinant types in selfing of F1 in dihybrid. Number of recombinants you do not get in F1. In F1, F2 generation you will get. In F2 generation you will get 6 plus 3 plus 3 is equals to 6 6 hybrids. Okay, all the recombinant types. Which chapters can I skip? I would recommend you not to skip any chapters. Just a second, okay? Children who are solving the question, just a second, let me answer. Otherwise, there will be huge trouble, okay? You should not be leaving any chapter. If you're planning to, I'll say the last 3 chapters, at least go through the questions. Do not skip it. Not skip it, just at least go to the questions once. Okay? Now, let's get into next thing. Okay, during pregnancy, the fetus develops limbs and digits after 2 months. 2 months means 1 month has 4 weeks. So 2 months means 8 weeks. After 8 weeks, my dear children, the fetus will be having limbs and digits. The same thing can be happening for other things also. So during pregnancy, the fetus has heartbeat. In which month? So you should know after 1 month. After 1 month, that is 4 weeks, heartbeat will be there. Now after 3 months, 3 months is not given, in the trimester is given. After 3 months, limbs and digits are completely formed, external genital organ, and most of the organ and organ system is also formed. After 5 months, the baby says, I want hair and I want to move. So baby will be having hair on the head and it will move around. 6 months, the baby will be having hair on its body. Eyelids will be separated, eyelashes will be formed. Okay, until 6 months you should be knowing. Please remember that. So your answer is B. Okay, Pratyush, Sumaya, everyone, Noor Fatima, all of this, great. Next thing, evolution. We are getting ahead with the next chapter, that is evolution. Let's get started. And over here, first question, theory of spontaneous generation was disapproved by experiments conducted by whom? Listen, what do you mean by theory of spontaneous generation? It said that, for example, I have a sack of wheat over there. One day I saw mice coming out of it. I thought from the sack of wheat a mice is made. Is that so? No, my dear children. So they believed it. That was theory of spontaneous generation. Now it was disapproved by someone who was what? I think female reproductive system will be coming. According to me, I don't know. It's a random guess. Okay, it's a random guess. Okay, theory of spontaneous generation was disapproved by Bia. Children, go through the question really well. Chinmaya says B, okay, Ammu says A, Pratyush says A, answer is A. It was Louis Pasteur. Louis Pasteur did his experiment where he had a thing like this. Okay, he sterilized this thing and it had a swan neck. Over here he had a broth. He found that this sterilized thing was kept sterilized only. There was no organisms up over here when it had a swan neck, probably because organism got stuck over here. But when the same thing was broken down and it was made like this, it was open and the broth was kept, other organisms came in and basically gave rise to some things. So he said life comes from pre-existing things, pre-existing life forms. Now that was also disapproved by Haldane and Operon later on. But theory of spontaneous generation was disapproved by Louis Pasteur, first guy. Okay, your answer is A. Going ahead with the next thing. The thorns of bougainvillea and tendrils of cucurbita represents what? Homology, convergent evolution, divergent evolution, or analogy? I'm very scared for the exam. Trust me, you do not have to be scared for the examination. You are going to do well. Just go at last moment. You do not have time to watch the videos. Just go through the questions, read it once. Okay? You are going to ace it. Please don't worry. So my dear children, thorns of bougainvillea and tendrils of cucurbita. These are the thorns and this is the tendril. You know what? Both arise from something that is known as axillary bud. So both has what? Same structure. Initially, at least, it has the same structure. So common ancestry is there for sure. And from them you get new things, my dear children. So this, basically they are evolved differently. So they are showing divergent evolution. Because they are in different habitats, but they have the same organs. They are evolved from the same organs, so they are homologous organs. So whenever you have things like this, my dear children, what do you say? HD vision, homologous organs and divergent evolution. So I'll say answer for this is 1 and 3. 1 and 3 are correct. So you have two things like that. Either way is correct. Okay, 1 and 3 is the correct option, not C. So this and this same option is there. So this is a typo error. It's the same thing. I hope you got it. It's B. Okay. Sharad, every one of you, please do not be worried. Just give your 100%. Trust me on that. Okay. Just your 100%. Nothing else we need. Okay. Charles Darwin in Galapagos Island found that finches had different forms of beak. This process of evolution is known as 3 A, B, C, D. Please tell me 3 A, B, C, D. 3 A, B, C, D. Oh, this is 2 and 3. I'm so sorry. So this is not 1. This B1 is the correct one. Thank you. Okay. Children, Charles Darwin, he went to Galapagos Island. He found that the finches which were there also finches were there, but all of them had different beaks. So basically, initially you had a group of finches, okay, they radiated to different places. They got adapted there because they had a beak which was able to take insects. That beak was able to eat, with that beak they were able to eat grass and everything. So this is known as adaptive radiation. They radiated and they got adapted there. So this is adaptive radiation. My dear children, this is an example of divergent evolution or convergent evolution. Tell me right now. I'm waiting for your answer. Okay, this is an example of divergent or convergent evolution. They are moving away, but they have a common ancestor. They do not have similar function now, but they had a common ancestor. What type of evolution is this? First, okay, I got your answer, that is A, B, C, D, but can you tell me what type of evolution is this? From one common ancestor they are moving away. Come on, champions. It is divergent evolution. Yes, they are diverging. Always remember, when you have a common ancestor, you're moving to different location. This is divergent evolution. Okay? Always remember, not convergent. Okay, now okay, I'll show you that question. I think you will get that question if you get it. Okay, listen, one very important question that I want to add up over here. If two organisms, okay, Australian marsupials, placental mammals, Australian marsupials, and you had placental mammals. Both of them were here in the middle region. From Australian marsupial category, one guy went there. Okay, one guy went here. So what is this? When you're talking about this alone, you say it's divergent evolution. But when you're talking about placental mammals also, both of them are here. From each stream, one guy is going to one location. So when they reach at one location, since there is a similar habitat, there are higher chances that they will be having some similarity among them also. So this is an example of convergent evolution. Both of them are converging at a point. So when two organisms are basically doing adaptive radiation in one particular location, you say this is an example of convergent evolution. You say this is an example of convergent evolution. Okay? But when you talk about it separately, that means you're talking about divergent evolution. Holy oy, I am a CS student. Then why are you here? Please go study CS. But I'm glad you are here, but wishing you all the best for your CS exam. But please go and study CS, okay? Children, I hope you got it. Children, please, 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 please don't spam over here, holy oil. Please don't spam. It's not good. Your fellow friends are studying, yeah? Okay, I'll just wait for finishing of this chapter. Okay, we just have 6 more questions, I think, and then we'll stop. Okay, a brown-billed duck is newly added to a farm of white-billed duck. This indicates what? A brown-billed duck was there. Okay, it was added to what? A farm of white-billed duck. There were so many white ones. In between that, you are adding a brown one. What is this an example of? Gene flow, genetic drift, mutation, or natural selection? This is known as, this is a factor that affects the Hardy-Weinberg principle. Yes, Noor Fatima, you're exactly correct. Together when you do, you call it convergent. Separately when they do, you call it divergent. So Chinmay says D, Gautam says B, not 2, fact 2 says it's A, Pratyush says it's A, answer is A, my dear children. It's known as gene flow. Okay, so here they had a frequency of alleles, but now from other places things are coming in, it is going to disturb the frequency, and now this is a factor that affects the Hardy-Weinberg law. So these all are the factors. Gene flow, genetic drift means all of a sudden, suppose all of a sudden by a random thing all the green ones are dying. So this is genetic drift. Mutation as in, for example, from the green ones there is some mutation and the red ones arise. Again the allelic frequency is disturbed. Natural selection as in, for example, there are red ones and there are white ones. There are white ones, but the white ones were able to sneak out when prey species were there. So it is natural selection, my dear children. Again, they will be increasing their number. Okay, so they are going to change the allelic frequency. Then there is one more method that is known as recombination. Recombination that happens in sexual reproduction where the chromosomes combine in different ways. Again, you get new combination, my dear children. Again, you get new combination. Again, making the Hardy-Weinberg principle go tata bye bye. What does Hardy-Weinberg principle say? Prajwal says, just a second, okay, Prajwal says, should I go to lunch and should I listen to, please have your lunch, okay. What does Hardy-Weinberg principle say? Hardy-Weinberg principle says that the allelic frequency will remain constant generation after generation. That means, suppose over here, you have capital T allele, that is for tallness, and you have small t allele. So frequency of this allele, my dear children, is going to be same generation after generation. So also small t, it is also going to be same. But can that be true? No, because you have all these factors affecting the generation, affecting the allelic frequency. So it not be same. Okay, the dominant alleles frequency is represented by P, the recessive allele frequency is represented by Q. The equation is P plus Q whole square. P plus Q whole square is equals to 1. 1 as in constant, it will remain constant. Okay, if you make it large, so it's p square plus 2pq plus q square is equals to 1. This is the equation. Ma'am, tell fixed diagram. Even the board would not know fixed diagram. And if someone is telling you the fixed diagram, that means the paper is leaked. Okay, so please do not believe any such people. Please practice all the things. Now, the biggest land reptiles that existed during Mesozoic era was Vishal, Hardy-Weinberg translated in randomly mating alleles and genotype remains constant even in the absence of factors. Yes. The allele frequency throughout generation to generation is constant, not same, you write constant. Okay. Yes, Arav, great children. So, tell me answer for this 5th question. The biggest land reptiles that existed during Mesozoic era was Ichthyosaurus, Tyrannosaurus, sea cows, crocodiles. Mehek, Zeba, there's new persons, I know also, someone new, hi children. So your answer is Tyrannosaurus. Your answer is Tyrannosaurus. The biggest land reptiles that existed during Mesozoic era was Tyrannosaurus. Okay? Okay. And also please remember during each era what are the organisms which are there in more amount. Now arrange the following in the order of evolution. If you heard me from the starting, you know the answer for this. In order, can you tell me what is the order? First came, I told you there were 2 guys, Ramapithecus and Dryopithecus. Dryopithecus more ape-like, Ramapithecus man-like. So this guy is the first guy. So C is first. So your answer is here. If you spot out C is first, you'll get the answer. After C comes B. After C comes Australopithecus. After Australopithecus comes Homo habilis, the Habibi. The actual hominid-like features, human-like features were found in Homo habilis. Okay, so from Australopithecus you got Habibis, Homo habilis. From Homo habilis you got Homo erectus. From Homo erectus you got Neanderthal man and Homo sapiens. So your answer is D. Yes, Ammo, Pratyush, Dhanush, Arav. Okay, not to fact, oh sorry, Vishal. Sorry, Vishwas. And Satya. Oh my god. Unique names. Okay, children. This we did know. Okay. In Miller's experiment, the environment of primitive earth was created and the temperature was. What was the temperature? You don't know my name? My name is Jasmine Joy. Okay? My name is Jasmine Joy. Yes. In Miller's experiment, the environment of primitive earth was created and the temperature was what? 400, 450, 800, 850. 50. Children, you're telling about 7th. Please put 7 and then write. So I saw Ammu who said 7. C, Dhanush C, Zaina C, Darshan, okay, 800. Kalkappa C, Hana also 800. Great. So children who are saying about C, you are absolutely correct. You are absolutely correct. It's 800 degrees Celsius. So there is a high temperature being maintained, my dear children. Okay. Prajwal, I'll take all the doubts together. Works? I'll take all the doubts together. Fine. Going ahead. The paleontological evidence for evolution is obtained from. Where do you get paleontological evidence? There are 4 type of evidences. 4 or 3? 4. One is basically this thing. What do you call it? Paleontological. Second one is embryological. Third one is comparative morphology and physiology. So please make sure you know all these things. Okay. So the first one, paleontological evidence was taken from. Easy question it is. 8th A. Chinmay says A. Ammu says A. Is the answer A? Sat, it's not 450, okay? In the previous question, it is 800. So, Arav, Sudha, everyone says A. Yes, the answer is A. So, paleontological evidence as in, for example, dinosaurs were there. How did we identify dinosaurs were there? When we went ahead deep, we found that between each layers, every time the earth is having multiple layers, my dear children, being formed. So, when the layers being counted, you saw that it's very down. So, that means they were evolved very first. And then after that, another layer was found. That means these guys who were found evolved from probably dinosaurs. So, fossils are evidence of evolution. Okay. Embryological evidence for evolution was disapproved by. disapproved by. Very important question. Question number 9th. So there was someone who said that no, whatever you're saying is wrong. Who was that person? So what do you mean by embryological evidence first? You know what, when we are embryos, when we are inside mother's womb, we have tail, we have gill slits, but do we have it now? No. So basically we passed probably through our evolutionary series. You know what, it is believed that first came Pisces, that is fishes, then came amphibians, then came reptiles, from reptiles came Ave, that is birds, and also us, that is mammals. So which is why when we are inside the mother's womb, we saw gill slits that is found in Pisces. Okay, we have tail that is found in probably amphibians or reptiles. You get it? Not amphibians, reptiles. Okay, so basically we are showing each evolutionary pathway. But some person said that this is totally wrong. This is totally wrong. B. So it is Carl Ernst von Baer who said that it is totally wrong, and who actually gave this theory? It was given by Ernst Haeckel. So both of them are Ernst, but the bigger name is the one who said no. So it is Carl Ernst Boy who said no. Why? Because in that case we should be showing all the characters of at least different characters of different organisms. We are just showing 1 or 2 characters, right? So that is wrong. Okay? Okay. Cool children. Which of the pair is either homologous or analogous? Tell me the answer. I am not able to solve MCQ, but I can do 2, 3, but I can't do MCQ. What should I do? The thing is, you will get MCQ if your concepts are strong. Okay, so at this point of time, I would not recommend you to obviously go through everything else. If you can, practice more MCQ questions. Okay, or try to understand what is the thing behind, the reason behind all the answers which are given. Don't just randomly blindly follow things. So you have to practice it. Okay, have the questions, practice it, give an error book. In that error book you write it, revisit it and rectify it. Okay, so I hope I helped you, Adarsh. And Hana says it's B, it's 9th. Children, 10. Arav says it's C, Kavita says I hope it's C, and Samiksha, Dhanush, everyone, A, B, C, D. Children, it will be homologous or analogous, either you want that. So homologous means they should be at least from a common ancestor. So sweet potato and tendrils of cucurbita, wrong. Okay, they are not showing any common ancestry, nothing, or they not. So tendrils, nor do they have same function. Wait, just give me a second. Homologous organs means the one that have at least the same ancestor. Okay. Analogous should be having same function. So sweet potato and tendrils of cucurbita, neither has both. Tendrils of cucurbita and sweet potato again do not have the same thing. Thorns of bougainvillea and tendrils of cucurbita, yes, it is an example of homologous because both arises from axillary bud. Both arises from the axillary bud, tendrils and thorns. Okay, so it is homologous organs. But sweet potato and wings of butterfly, no. Wings of butterfly, no. So your answer is C. Your answer is C, my dear children. Okay, children, before I get into the next slide, okay, answer is C. Can you give me an example? Can you give me 3 examples of convergent evolution? 3 examples of convergent evolution. This is the last question of evolution chapter, okay? And by then you think about 3 examples. So observe the following diagrammatic representation of operation of natural selection on different traits. Here the natural selection depicts what? So there are certain graphs given in your NCERT. Okay, one graph looks like this, other graph it looks like this, and the other graph it looks like this. So this is what? So Chinmay says butterfly and wings of birds are okay convergent. Okay, sweet potato and potato, wings of insects and birds, great. And Australian marsupials and placental mammals, oh my god, great my dear children. Okay, great. I'm just going back for a second. Okay, listen, wings of birds and butterfly, bird is basically bird and butterfly is an insect, but both of them have wings for flying. Structure is not same, so these are analogous organs. These are analogous organs and they show convergent evolution, A, C and H, D. Okay, analogous organs, and they show convergent evolution because they are in the same habitat. Sweet potato and potato showing convergent evolution, but they are still analogous organs. Why? Because they have the same function of storing things, but potato arises from stem and sweet potato arises from root, so origin is not same. So again they are convergent together at one point, but they do not have common ancestor. Now the eye of octopus and of human beings, eye of octopus, octopus is invertebrate, human beings are vertebrates. Both of them use the eyes for seeing, but not common, no common ancestor. Again an example of this. And also, as I said, this thing, Australian marsupial and placental mammals. Okay. Okay, now over here. So, oh my god, I told you the answer. Okay, so this one is stabilization, okay, where what is happening? Suppose there is two characters, tall and short. In between, if organisms, or basically nature is choosing organisms having the median character, that is middle height. So this is stabilizing thing, okay. Now over here, if it is choosing one extreme, either tall or short, this is known as directional because it is moving to one direction. If it's choosing both the extreme traits, it is known as disruptional. So over here it is disruptional, that is given. Okay, so 11 is C. Fins of sharks and fishes is a wrong thing. Flippers, okay, flippers. Flippers of dolphin and probably penguin. Flippers of dolphin and penguin, they are again showing analogous organs and convergent evolution. So dolphin is a mammal and penguin is a bird. Both of them are going to use their flippers for swimming. So it is an example of very important question. Please know about analogous and homologous organs. An example, if you are not knowing it, you are going to mess around your things. Okay. Prajwal, so thankful. Okay, oh sorry. I think this one more question is there. How many questions are there? Children, I'm so sorry. There are two more questions. Okay. If diagram has an arrow mark, it is directional, and no arrow mark stabilized and disruptive like mountains. Okay, so listen, sometimes they would not be giving you arrow marks like this. They will just show in one end it is going, so it is directional. If it is in the middle, it is stabilizing. If it has two mountains, so basically it is disruptive. Disruptive as in it is broken down, right? Now, the factors that affect the Hardy-Weinberg equilibrium are listed below. Which of them is this answer? Factors are given, you have to tell which is the correct option. So, crossing over and independent assortment. What do you mean by independent assortment? Independent assortment means you are getting new character. I told you, independent assortment happens, that's why you get a new character. And Hardy-Weinberg says there is no chance of new character. The allelic frequency are going to remain the same. So, this is one reason that affects the Hardy-Weinberg. Crossing over results in genetic recombination. So, again recombination as in this is also one reason that affects, right. So, crossing over and mutation also affects, again mutation also affects, genetic drift also affects, everything affects. So, I will say 1, 2, 3, 4, all the options are correct, all of them are going to affect Hardy-Weinberg principle. So, answer is D, not A, it will be D. Why did he say A? What is the reason behind A? If you tell me the reason behind A, I will approve. I opened some other video. Okay. It will be D. I hope you understood, children. Children, have you understood? If understood, can you just give me a heart or a thumbs up? Okay, if understood, just give me a heart or a thumbs up. I'll understand that you understood. If no one gives, probably I'll think you didn't understand. I'm waiting because A is A. Because A is A. Okay, got it. Thank you. At least someone said, okay. Ammu, I hope Noor Fatima, okay. Thank you, dear children. Okay, again, this question we have solved, okay, previously. Okay, P plus Q whole square is equals to P square plus 2PQ plus Q square represents an equation used in population genetics, molecular genetics, Mendelian genetics, biometrics. Oh, so many hearts. Thank you. Ma'am, have a lunch break. I'll take. So considerate. I'm so glad to find children like you who are thinking about teachers also. 14th A is population genetics, molecular genetics, Mendelian genetics, biometrics. Yes, it is population genetics. Okay, it will be population genetics. Okay, and this is Hardy-Weinberg's law. Please make sure you're writing 1. 1 means constant. Okay. Can I tell you one thing, one very important thing? P square means you are talking about how many organisms are tall or basically have the homozygous trait. Okay. This means how many organisms has, this is Q square, this is P square. So if you're given the value of P, my dear children, for finding capital, the homozygous thing, what do? You do P square. Okay. If you're given the value of recessive allele T and you are asked how many organisms will be, what is the frequency of small t, what do you need to do is that do Q square. Okay. Always remember that. And if you are asked to find what is the frequency of organism having capital T, small t, that is heterozygous individuals, how do you find out that? You write, when you open this, you get this right. So you write 2pq. 2pq suggests that. So 2, whatever p's value is there and whatever q's value is there, together when you do, you get capital T, small t's frequency. Okay. Suppose you are just given A. Okay. You are just given A. What do you need? Sorry, you're just given one frequency, that is P's frequency. How do you find Q's frequency? You do this. P plus Q is equals to 1. Over here, don't put the whole thing. Just for finding Q, I'm telling you. Just for finding P or Q. Suppose this is given as 0.4. So what is Q? 0.4 plus Q is equals to 1. So Q is equals to 1 minus 0.4. That is 0.6 is Q. Okay. Rest of the time, this will be the equation. Always remember it. Is this formula important? Very much important. Very, very, very much important. Okay. Yes. So from this point onwards, I'll take few doubts and then only I'll be going ahead with MCQs. Okay. Since a lot of children asked about doubts, P is equals to 1 minus Q. Yeah. Okay. But the formula usually is P plus Q whole square is equals to 1. That is what we usually take. Okay, ma'am, chalo, ma'am, please have lunch and we will also have lunch, otherwise you will, please, you can go and have lunch, no problem, okay? I will be, because you know what, I have to record one more video for you, that is 30 minutes revision video, and I have to, so there are a lot of things you want to understand. So it's okay, thank you for considering me. My mother tongue is Malayalam, I am a Malayali. Okay, so microbes in human welfare. Should we start it or do you have doubts? So someone was asking about Michelson-Stahl experiment. That someone is here. The person who was asking about Michelson-Stahl experiment, are you here? If you're here, can just give me this thing. No? Some other doubts? Please explain cycle of Plasmodium. Okay, Plasmodium cycle, since first come first serve. Plasmodium, listen to me. So we have mosquito. Okay, inside the mosquito's salivary gland, the mosquito has plasmodium. Okay, in the salivary gland of the mosquito, the plasmodium is there, and the stage in which it is there, it is known as sporozoite stage. What stage? Sporozoite stage. Where is it? In the salivary gland. Now the mosquito comes and bites us. When the mosquito, it comes and binds us. Okay, what will happen? It does not give blood. It has come to take blood. It releases saliva. Because in the saliva it has certain enzymes which basically anti-clots a lot of things. So these enzymes are given out, and along with it the sporozoites also get into the human body. So in the human body, in which stage is the plasmodium going to enter? Sporozoite stage. Very important question. Now in the human body it has two camps. First camp is liver. It is going to affect the cells of the liver, my dear children. It is going to convert into different stages. It's not just going to stay in sporozoite stage. In the liver, they will convert into stages, they will divide themselves and they will rupture, they will open the cells of the, liver cells, which are known as hepatocytes. Once they open, this is going to the next camp, that is RBC or erythrocyte camp. Now in RBC, my dear children, what happens is that this particular plasmodium does two things. Okay, one is basically to break down hemoglobin to heme and globin, and it converts heme to hemozoin, because if it stays in heme state, what happens? It is very toxic for the organism, plasmodium. So it converts into hemozoin state in the hemoglobin, in the RBC. So in the RBC you have hemozoin being made. Apart from hemozoin, what also happens is that it converts into a stage which is known as gametocyte stage. gametocyte state. So two things happen, hemozoin formation and gametocyte state formation. Now the RBC is ruptured, the RBC ruptures open. When it ruptures open, what will happen? The gametocytes are released and hemozoin is released. Hemozoin is going to cause chills and fever. Chills and fever. What is going to happen with the gametocyte? The gametocyte which is there, it is just there in the blood. It will just release in the blood. Again, another female Anopheles mosquito is going to drink this blood. Female Anopheles mosquito is going to drink this blood. When it drinks the blood, what will happen? The blood goes to the gut, and in the gut, the gametocytes are entered, right? The gametocytes are going to form gametes, are going to form gametes, and the gametes will be fusing in the gut itself. In the gut itself, they will be fusing, and then the fused thing, whatever is formed, it is known as eukaryotic. Eukaryotic will slowly go to the salivary gland again and form the sporozoite. That is the cycle of Plasmodium. I hope you got it. Okay. Please tell about Miller's experiment, independent assortment, linkage and recombination, exam point. Okay, coming one by one. Okay. Bioactive molecules. Okay, coming to that also. So first, I hope this is done. Plasmodium is done. You have three things: Plasmodium vivax, Plasmodium falciparum, and Plasmodium malaria. Plasmodium falciparum is the malignant one. Okay, this is done. Going ahead with the next thing. We have second person who asked was about Miller's experiment. Okay, so Miller's experiment, let's talk about that. Okay, Halden and operon was there. Okay, and operon was there. So they said that basically we organisms, or basically organic molecules that you see today, have come from inorganic things. And these organic things which are made, okay, just a second, the organic things which are coming from inorganic things, and this organic thing which is made, it forms up different life forms. Okay, I'm not able to write. Anyways, that was said by operon and Halden. And when it was happening, my dear children, remember two things were also there. That is, there was a reducing environment, that means there was no oxygen, and also the temperature was really high. So there came Miller, who basically said that I will experimentally prove it. So what he did that, he had an experiment where basically he set a closed setup. There was nothing else that was getting in, okay, and he had a broth wire where, just a second, okay, what happened? I think there's a time for glitch. The board is tired of me.